Okay. Shall we get going, Sarah?
You are welcome to begin.
All right. And why don't you go ahead maybe and stop sharing the screen and I'll share mine. Well, hello everyone and welcome. My name is Ken Cornetta and I welcome you to the Clinical Gene Therapy ASGCT Indo-UK Symposium. The goal of our symposium today is to look and have discussions from speakers from the UK and India who are looking to develop and share their experience and insights onto factors that will be important to initiate clinical trials of gene and cell therapies in India. This hopefully will stimulate more clinical research, and we feel may be an important step to expanding the availability and access to these therapies to patients in the area. Just a note, at the end of each session, the speakers will participate in a panel of Q&A. The audience, folks, are really encouraged to type questions into the Zoom Q&A feature throughout the presentations. You may continue to submit questions also during the panel discussions where we'll attempt to answer the questions that are raised. Before we begin, I'm just going to present just a couple of slides for a little bit more information. And just sort of where this was, this symposium grew from. And it was from the ASGCT, and part of our mission, which is, as sort of noted here, to advance knowledge, awareness, and education, and the hope of leading to discovery of clinical applications in genetic and cellular therapies. But it's all about alleviating human disease. And to do this, it's a pretty big lift. I am at Indiana University, and for over 20 years now, we've been looking at manufacturing vectors for clinical gene therapy trials. And so we have some of the things here at Indiana, but truthfully, we're now focusing on cellular therapy. But when we're developing a gene therapy product from an academic perspective, we really need to engage basic scientists, clinical investigators. We need to sort of do that preclinical work to find out the appropriate vector technology. And then we have to manufacture that in a way that's suitable for human use. And particularly for the area that I work in, which is retroviral and lentiviral vectors, cell therapy is an important part too, because we generally take cells out of the body, introduce the vectors into them, hopefully correct their genetic disease or give them functions such as anti-cancer functions. And we have to do this in the setting of a clinical trial, and we need to do this with hospital support. And there's many steps along the way, and it can be very challenging, but I think there's many interests, and this is a shared interest, both at Indiana, throughout the US, and throughout the world, of how do we bring these innovative therapies to the patients that we see. Now, pharma has been doing that, and part of the challenge is some of the price tags that pharma has charged for some of the products that have been approved for gene therapy have been extremely expensive. And even in the States, because of the way our insurance compensates, this still is out of the reach for many patients to be able to receive some of these therapies. And so we're thinking about ways, how do we try to maybe do this more locally? And I think there's been interest, and certainly the folks who've helped organize this, which has been our committee at ASGCT, and actually a number of these really critical folks on that committee will be speaking today to you, is how do we sort of share our knowledge so that we can reach the most patients? And part of this really started in 2020 when the ASGCT strategic plan specifically outlined global outreach as one of the things that we wanted to do. And that's when our committee was formed. We've only been working over about a year. And we're trying to, again, foster equitable access to gene therapies all around the world. There are a couple of things that the societies did, and I want to just make you aware of some of them. There's a number of online resources at asgct.org. about cell and gene therapy. There's actually some patient journeys of folks who have been treated with some of these modalities. And if you want to see what kind of clinical trials are ongoing, there's a list of those on the website. And there's also some professional development resources there too. The other thing that we did was to try to get more folks knowledge about what's going on in gene therapy. We've lowered our dues to join the society. For low and middle income, we had a 75% reduction. And if you're in a lower income country, the cost is only 10% of what it would be for a high income country. So hopefully folks can get more from this. We've been doing symposiums. Our first one was in Brazil. And again, we met with folks there, also in South Africa. And now again, from some of the speakers you heard today, to say, what do you think are some of the pressing issues to bring cell and gene therapies into the mainstream in India? And so we're going to hear about some of those efforts today. We have some other initiatives that we're looking at. One is educating more about gene therapy in lower and middle income countries, but also looking at an educated program for the challenges of conducting gene therapy trials. They have some certain specifics that need to be addressed, and we're hoping in 2022 we will have at least a day to 2 day long training for folks who are interested in doing clinical trials and what may be required. And we're looking to develop a faculty development program where we can actually have direct mentoring of folks with other folks in society members who want to bring gene therapy both at the basic science and the clinical arena into fruition. So leave this up for a second, but we'll hear more about that. And really looking forward to an exciting afternoon of talks around cell and gene therapy. Thank you very much. And we'll go ahead and let our next speaker and our moderator, Dr. Savita, please welcome.
Thank you, Ken, and a warm welcome to one and all, and good day to you. My name is Dr. Savita Rangarajan, and I'm an Associate Professor of Clinical Hematology at the University of Southampton's Faculty of Medicine. I've been a hemophilia treater for more than 2 decades and have been part of many gene therapy trials. I also run a clinical trials and research unit at KJ Somaiya Super Speciality Hospital and Research Center in Mumbai. Before we go on to the session, I will take a moment to thank ASGCT and the sponsors Park Therapeutics, and of course, our knowledge partners and collaborators in making this virtual meeting happen. And without much ado, I would like to introduce our first speaker of this session, Dr. Jayendran Rao. Dr. Rao is a professor and a DBT senior fellow in the Biological Sciences and Bioengineering Department at the Indian Institute of Technology, Kanpur. He has spent years on working on AAV vector biology and has many publications and patents to his credit. Dr. Rao is also serving as a co-chair with me for this event. Dr. Rao, over to you.
Thank you, Dr. Savita, for the very kind introduction. And it's my pleasure to introduce our work to the audience. Over the next 20 to 25 minutes, I'll give a broad stroke of some of our initiatives on preclinical gene therapy in India. So for the sake of students, the term gene therapy refers to the introduction of a normal copy of a gene into a patient with a genetic disorder so as to replace the missing protein function. The most important component of gene therapy is the vehicle that is used to target specific tissues in the patient. There are several types of vehicles available, or they are non-viral and virus-based vectors. Among the virus-based vectors, there are multiple components such as the lentivirus, retrovirus, and parvoviruses such as adeno-associated virus. And generally for gene therapy, viruses are preferred because they have evolved naturally over thousands of years to target specific organ systems in the human body. And today I'll be focusing on a parvovirus, the adeno-associated virus, commonly abbreviated as AAV. AAV was first described in 1965 as contaminant of adenoviral culture. The virus is very minimalistic. It contains two genes, rep, that encodes for the replication-dependent function of the virus, and the cap that encodes for this icosahedral capsid. If you look at the virus, it is one of the very smallest viruses, about 20 nanometers in diameter. And the biggest advantage of using AAV as a gene therapy vector is its relatively non-pathogenic nature. And in case of recombinant AAV, we get the wild type genes and replace it with our gene of interest so as to use this scaffold to target specific tissues of interest. Basically, we are using this as a Trojan's horse to target specific tissues in the human body. At least 10 common serotypes of AAV are known from AAV 1 through 10, and each one of them have a unique sequence and structure that gives them the ability to target multiple tissues in the human body. For example, if I would like to target the eye, then I would use AAV2 serotype. For the case of liver, I would use either AAV5 or AAV8. And if I would like to use, let's say, for a hematopoietic system, then I would use AAV6. Almost most of these serotypes have been used in various preclinical as well as clinical studies. While in preclinical studies, our experience has been very good. Most of the therapies seem to work. But when it comes to phase 1-2 clinical trials, there have been definite successes, such as in the case of hemophilia B, as well as in Leber congenital amaurosis. Hemophilia B is a bleeding disorder. I'll talk more about it. And Leber congenital amaurosis is a retinal dystrophy. However, there are few key challenges, such as the long-term efficacy of the gene therapy product, the immune response from humans against these gene therapy products, and more importantly, in the context of today's talk, the access to these GTPs, particularly in lower and middle income countries. Let us first start with hemophilia. What is hemophilia? It's a bleeding disorder. And why does it happen? When you have a trauma, normally there is primary hemostasis where the circulating platelets will come and bind to the breach portion. And this platelet plug is further reinforced by the fibrin clot. And the formation of fibrin involves a series of enzymatic reactions on the surface of platelets. And these enzymes are serine proteases, or otherwise called as coagulation factors. In the case of hemophilia, when either factor 8 or factor 9 is deficient, the patient can no longer form the fibrin clot. As a result, they have a tendency to bleed into soft tissues and joints. So based on the residual amount of either coagulation factor 8 or factor 9, patients could be classified as having severe hemophilia or any one of the other two forms, which is mild and moderate. Recombinant protein replacement therapy is available. However, they are pretty expensive. Gene therapy is considered to be an attractive option because not only it is curative, but also has a wide therapeutic index. So for example, a patient converted from being severe hemophiliac to moderate and mild hemophiliac is a big jump in terms of phenotype because they would bleed only post-traumatically. So there have been several clinical trials for hemophilia. This is just a snapshot of some. I'm not getting into the details, but just suffice to say that majority of them have shown promising leads, particularly with these two modes of therapy, which are AAV based, and they have showed almost near physiological levels of factor 8 or factor 9. But the catch is the pricing. Now, hemophilia gene therapy is expected somewhere in the 1 to 3 million range, but I suppose it would be somewhere closer to the 1 billion range. And Dr. Savita will probably throw more light on her talk on this. And this is beyond the reach of many, particularly in developing countries. The second disease that I would like to highlight is about Leber congenital amaurosis, which is a retinal dystrophy. And this is caused due to mutations in a gene called as RPE65. Now, RPE65 is an isomerase enzyme, which is present in the retinal pigmental epithelial layer within the eye. In case of patients who carry mutations in this RPE65 gene, they can no longer convert the substrate 11-cis retinol to all-trans retinal esters. And therefore, the accumulation of the substrate leads to the degradation of the RPE layer, which can no longer support the photoreceptors or nourish the photoreceptors. As a result, there is a vision loss that leads to Leber congenital amaurosis. Gene therapy has been tested using AAV vectors, particularly of serotype 2 via subretinal administrations. While the initial data is extremely promising, the patient's vision was rescued. But after a longer follow-up, it was realized that beyond 1 to 2 years of gene therapy, the phenotypic manifestations that had improved went back to the baseline. So this calls for developing next generation of AAV vectors, which are ideally having improved transduction efficiency, reduced immunogenicity in the host, and has the ability to bypass pre-existing immunity, because AAV is quite prevalent across the population, and therefore it should have the ability to bypass pre-existing immunity in humans. And more importantly is the cost factor, which has a major impact as a pharmaceutical agent in lower and middle income countries. Now, if you look at the gene therapy products that are currently available in the market or being developed, the majority of them have been developed in the United States or in the UK. You can see the distribution of a number of gene therapy products that are under development. And they have been developed for a variety of conditions. But when it comes to LMICs, given the fact that a technology like this requires significant investment in research and development, and there has to be adequate know-how and competency, and also the regulatory frameworks needs to be established, because once you have the product, how do you take it beyond just testing them preclinically? So these are some of the important factors that currently is not optimal in terms of LMICs. So why is it useful to develop them, first of all, in LMICs such as India? Now, generating local GTPs is crucial, because not only can you improvise the efficacy, but you can also manufacture them in adequate quantities and deploy them to the patients in need. And more importantly, if they are generated locally, it creates an enormous value addition and cost advantage to the patients. Secondary to that is the requirement of clean manufacturing facilities for vector production and also the scale up. And typically they are mostly available to recombinant proteins or cell and blood-based products in developing countries. So the main motivation here is that to develop indigenous vectors and also to scale up these technologies for GMP grade vector production, so as to reach promising mode of therapy to a larger proportion of patients that are in need. So in this initiative, I'm just going to broadly show you what we work on. So we started this work a few years back, trying to understand the biology of the virus and the host cellular interactions so as to develop local and indigenous GTPs. To do this, we study the biology of interactions between the virus and the host cells at different levels so that we can identify rate limiting steps which are inhibitory to successful transduction with AAV viruses. Now, understanding this rate limiting steps is crucial, then you can deploy strategies to overcome them. I'll give you some examples of our work, particularly in case of hepatic gene transfer, ocular gene transfer, and suicide gene transfer, just a snapshot of data and some of the data that we have generated preclinically in animal models. The first approach we took is what is called as capsid bioengineering. And when a virus such as AAV infects a cell, it has to go through multiple steps before it enters into the nucleus. And our hypothesis was that given that there is a huge exogenous load of viruses that is directed against a target cell, the cell recognizes them as foreign, uses the kinase machinery such as serine threonine kinases to code the capsid-based amino acids, particularly at serine threonine residues. Now, once this marking happens with phosphorylation, the ubiquitination happens because the ubiquitin proteasome system can degrade the viruses. So typically what happens is when you use 100 viral particles to target any given cell, less than 30 make it to the nucleus, and close to 70% are degraded into the cytoplasm and are not available for gene expression or for protein production. To reverse this, we thought that we should identify those residues that are specifically marked by phosphorylation or by ubiquitination. To do this, we took the structural bioinformatics approach. We studied the AAV2 capsid, and we identified what is called as the phosphodigrons. Now, what are these phosphodigrons? They are serine and threonine phosphorylation sites recognized as degradation signals by ubiquitin ligases. We identified 3 such phosphodigrons on AAV capsid, and once we identified them, the next step was to mutate residues in and around these phosphodigrons, particularly those containing serine, threonine, and lysine residues. So now you had a wild type virus and modified versions of this virus. And the next step was to test if these modified or mutant viruses were effective. To do so, we used reporter gene, that is GFP, packaged with either the wild type virus or the modified viruses. And I'm not getting into the details of the data, just to show you that some of the mutant vectors that we developed had 2 to 3-fold increased gene expression, particularly when we measured GFP by flow cytometry. The same, when tested in animal models by in vivo hepatic gene transfer, showed roughly a 3 to 4-fold increase in gene expression. Now, this was with AAV2, which is the prototype virus that we started off with. And when we look more closely, many of the residues that we started mutating in AAV2 serotype were also conserved across all the 10 serotypes, which meant that these could be potential sites for modification, and we could reap the benefit of modifying them and deploying them for other tissues. So the next stop was AAV8, and as you can see here, these were animals that were administered with unmodified AAV8 wild type vectors. This is the level of luciferase or the transgene expression 2 and 5 weeks after hepatic gene transfer. And these are some of the mutants that shows roughly 2 to 3-fold increase in luciferase expression. We then used the therapeutic gene, which is human coagulation factor 9, and then tested some of these mutant vectors in hemophilia B mice, transgenic mice. And as you can see here, we were able to see near physiological levels of factor 9 production in those animals that were administered with AAV8 mutant vectors. So this was the first part. The second part, and the more important component, is these viruses or the vectors, recombinant vectors, when they are packaged, they typically use a human cell line, which is HEK293 based. And during their production, they are likely to acquire post-translational modifications because they have to use the cellular protein synthetic machinery. Details on some of them were known, such as phosphorylation, acetylation, and ubiquitination, but the effect of other post-translational modifications. What is a PTM? It's a chemical modification that builds in variability and diversity to the protein. So the effect of other post-translational modifications on AAV capsids were unknown. And again, I'm going to cut a lot of data. We focused on 2 particular types of post-translational modification called as summoylation and the other called as nedylation. So these are basically post-translational modifications where a specific chemical moiety is added and the protein is marked for subsequent degradation. Now, we created a repertoire of multiple mutants, and I'm going to show you data from only one of them, one of the summoylation mutant, which was a change from lysine 105 to glutamine, and same for lysine K665 to Q. And this is the data that we obtained after hepatic gene transfer in hemophilia B mice. And after hepatic gene therapy, we could see that both the nedylation and the summoylation mutant produce supraphysiological levels of factor 9 at the doses that were administered in hemophilia B mice compared to the wild type vectors and the untreated controls. And it was also important to ascertain if these mutant viruses or vectors that were used for hepatic gene transfer were immunogenic. A limited ELISpot assay showed that these vectors were indeed non-immunogenic in the recipient mice. So to summarize this part of the work, hepatic gene therapy with phosphodegron or nedylation and simulation site modified AAV vectors demonstrated physiologically relevant factor 9 expression in animal models of hemophilia B. So once we initiated this work, then the obvious question was, would such a vector be useful for other diseases? And because we were focusing on ocular diseases, we tested some of these mutant or the novel vectors that we developed in a retinal dystrophy model. So this is basically a mice called as RD12, which is deficient in RPE65. And as a result, we packaged the therapeutic gene, that is RPE65, with some of these mutant vectors, and a host of parameters were titrated. And just to give a snapshot of the data, so this is eyes that received the mutant vector containing GFP as the transgene, and this is the wild type vector. At similar exposure, you see roughly about 8-fold increase in gene expression. This was with the marker gene or the reporter gene. And when we repeated the same experiment with the mutant vectors containing the RPE65 gene, which is a therapeutic gene, in LCA2 mice, this was the data that we obtained. You can see that these are the results of electroretinogram analysis. This is untreated mice, which were dark adapted. And when you give a light impulse, you do not see any photoreceptor activity, the A wave or the B wave. When you administer them with wild type vectors, yes, there is some phenotypic rescue. But when you use the mutant vectors containing the RPE65 gene, you see a clear A wave, which signifies a rod cone response, and you see a clear B wave, which signifies the bipolar cell response. So once we had these mutant vectors, the next obvious question was, how can we improvise further on them? So when we package these viruses, I mentioned to you that we use a producer cell line, which is of human cell origin. And typically, we harvest these viruses that are packaged within the cellular compartment, which are intracellular. More often than not, the extracellular media is considered as a waste and thrown away. And in a few years ago, these extracellular media were reported to contain extracellular vesicles that also contained AAV vectors, and they had been shown to be very efficient in terms of transducing the eye. Now, given that we were working with the mutant vectors, our obvious question was, if we use the mutant vectors to package similarly, will the exosome carrying the mutant viruses have a better phenotype? And this is just a limited data that shows that the mutant vector containing exosomes had wonderful gene expression in the eye when compared to either the wild type vector or the untreated animals. So this part of the work is currently undergoing validation with the therapeutic transgene in LCA2 mice models. And this is the data only with respect to the reported gene constructs. So what have I told you so far? Molecular engineering of AAVs at specific post-translational modification sites. With this, we have been able to show better phenotypic response in terms of hemophilia B mice or in a LCA2 mice model. Exosome-associated AAV vectors seem to be promising, but require further validation. I'm going to change gears and talk about one more approach that we have done preclinically. In the last few slides, I had talked about replacement gene therapy, where the intent was to replace the missing protein function. So therefore, we had replaced it with a normal copy of the transgene. Herein, the idea is to kill the target cell because the target cell happens to be a cancerous cell. And therefore, we developed optimal suicide gene therapy approaches using AAV as the delivery system. And the disease that we targeted was hepatocellular carcinoma, which, as you know, is one of the most common primary liver malignancies and has a leading cause for cancer-related deaths in patients worldwide. While there are chemotherapeutic agents like sorafenib or surgical resection possible, typically they are not useful in end-stage disease or when there is resistance to the drugs. So always having newer strategies would be helpful. So given that we had hepatotropic AAV viruses, and given that there was inducible caspase 9 described, which was used as a T-cell safety switch, we use this system to kind of target these cancerous cells in terms of hepatocellular carcinoma. So what does this virus contain? The virus encodes the inducible caspase-9, which when it expresses the protein is expressed as a monomer. This monomer needs to be dimerized so that it can activate the downstream caspases. And for this activation step, we use a chemical inducer of dimerization or a drug called as AP20187. And when we employed this, it leads to the activation of downstream caspases such as caspase-3, leading to the target cell apoptosis. Again, a lot of developmental work happened. I'm not getting into the details. What I'm going to show you here is just the in vivo data. As you can clearly see here, that those animals where this is a xenograft model of hepatocellular carcinoma that received AAV vectors containing the suicide transgene, you could see that there was a clear tumor regression compared to those animals that did not receive the treatment. However, this by itself did not eradicate the tumor completely, and therefore further optimization in terms of the transgene or the level of expression needs to be taken care of. And this is pretty much a work in progress. So the same data is recapitulated. This is the apoptosis data in the target cells within the tumor of those animals that were treated with this inducible caspase-9 versus those that were not treated with them. So to summarize this part of the work, we developed AAV-based inducible caspase-9 system to target hepatocellular carcinoma, and that shows clear benefit of using this system. A similar strategy was undertaken with AAV6 based exosomes, and they were also shown to be efficient. So what have we done? And so we have developed multiple technologies based on AAV. And today I've given you a very brief snapshot of the data in hemophilia mice models, in murine models of Leber congenital amaurosis and hepatocellular carcinoma. What I have not shown, but I have also worked on, is in acute myeloid leukemia and lymphoma. So we have developed technologies spanning the vector capsids, the transient constructs, and also the vector production processes. So what are the next steps for us? Given that gene therapy is a very nascent field in the country, the first and foremost requirement was development of regulatory processes. And Dr. Geeta Jotwani, who will be speaking in one of the next sessions from Indian Council of Medical Research, organized a team of experts, and we have completed this as of 2019. And now there is a clear path to move towards vector production and clinical trials. And currently we are in engagement with industry partners for GMP production of our AAV vectors. And some of them are also getting licensed so that we can actually start or initiate clinical trials. In terms of clinical trials, we are in touch with a couple of clinical partners for carrying out the phase 1/2 study, both in terms of ocular diseases such as LCA2 and also for coagulation disorders such as hemophilia. Finally, I would like to thank my team and also my collaborators for their help and support, and my funding agencies for their generous support. Thank you for your kind attention.
Thank you, Dr. Rao, for your talk. And we will take questions after the next speaker. So let me introduce Professor Tuddenham. Professor Tuddenham is synonymous with hemophilia. He's an emeritus professor of hemophilia and a consultant hematologist at the University College of London. He was responsible for purifying and cloning the factor VIII gene, which led to the highly effective recombinant factor VIII concentrates, and is a pioneer for hemophilia gene therapy. Welcome, Professor Tuddenham will be available for Q&A much later, but we will be playing his talk just now. Thank you.
Freeline Therapeutic Limited, a spin-out company which is devoted to using AAV as a platform for gene transfer in hemophilia and other monogenic disorders. Gene therapy for hemophilia has a history going back to the last years of the previous century, but the first successful trial in terms of long-term sustained factor level began at the Royal Free in 2010. So I want to cover a bit of what we've learned in the subsequent 11 years. In the left upper panel, you see the most significant results of the Avogen-sponsored trial in which a subject was infused with an AAV2-based factor IX vector directly into the hepatic artery. He attained an impressive rise in factor IX level, which, however, was short-lived because in the left upper panel, you see the most significant results of the Avogen-sponsored trial in which a subject was infused with an AAV2-based factor IX vector directly into the hepatic artery. He attained an impressive rise in factor IX level, which, however, was short-lived because, coincident with a rapid rise in the liver enzyme alanine aminotransferase, the factor IX level fell. The patient remained well. This was a subclinical event, but it was attributed correctly, as it turned out, to an immune response to vector capsid. Building on that study, we, and I mean my colleagues at St. Jude, and in particular Amit Nathwani, who became my successor, developed a new study in which the vector consisted of a self-complementary expression cassette in AAV8, which was able to be given by peripheral vein infusion. We used a stepwise increase in dose, and at what we then term our high dose, which is shown in the right-hand panel above, participant 5, after this infusion, at a dosage of 2 × 10⁻12 vector genomes per kilogram, rapidly achieved a level of factor IX in the range of 5 to 10%. However, after 8 weeks, the evidence of liver inflammation was obvious, the ALT rose, and the factor IX level fell. We started a course of prednisolone, but perhaps a little late, and his factor IX level actually fell as the ALT level fell, and has settled out around 2 or 3%, which persists around that level subsequently out to about 10 years. We were, with the next participant, perhaps fortunate in that his ALT did not rise so rapidly, but we were able to give a short course of prednisolone to control that, and although his factor IX level had fallen to 5%, it subsequently rose and has settled out around 6%. And I can now state that 10 years later, it's still at that level, and he has not needed any further factor IX treatment for spontaneous bleeds. He's had a few trauma-associated bleeds, but he has clinically turned from having a severe type of hemophilia B to a clinically mild. Now, we published a follow-up on the study of the first 10 patients in 2014, which was considered a landmark, and we had at that point data up to 4 years with a consistent, durable response and no lasting adverse effects. Liver enzyme elevation was controlled, and the bleeding rate was reduced substantially in most of those patients, actually to near zero. I should point out that when you look in detail at these individual patient results, there is considerable variation both in the level achieved and in their immune response. 2 of the subjects had no immune response. This is a pattern which we see in subsequent studies, that the immune response is a big variable and the personal response is also a variable. And we don't understand yet fully the underlying causes of this variability. I would like to just draw attention to an excellent review by Perrin, Herzog, and Marcusic, which appeared in Blood 2019. And I've filched a slide here with shameless plagiarism, but acknowledging it, which shows what can happen in terms of the immune response when we use adeno-associated viruses as vectors, which are highly efficient at transferring foreign DNA, of course, in nature their own, into a target cell. And of course, equally, our immune systems, particularly the human immune system, is highly evolved and developed to resist such attacks, which is why if there is an immune response, you can end up with a loss of efficacy, whereas if you can control or there's no immune response, you get a lasting effect. I commend this paper to those who are interested to study this subject in further depth. The challenge is to reach a normal factor IX level. That is understood by all in this field to mean a cure. And a cure is normal hemostasis for life after the treatment. But the challenge is met now by using a variant that was discovered in a family in Padua by Paolo Simeoni and colleagues, in which a minor variation in the factor IX sequence, a single amino acid change, leads to increased catalytic activity. So for each nanomole of factor IX expressed, you get 5 to 8-fold more activity than with the wild-type sequence. Two studies in the literature report on this, and the third study I'm going to refer to is our own, using this mutant. And there's been quite impressive data in two studies, one from Spark and one from Unicure, in which patients have now attained impressive elevations of factor IX, well above those that we observed at our original study, simply because the actual factor IX excreted from the transduced liver cells is 5 or 8-fold more active. There is other approach possible apart from increasing the potency of the protein expressed, which would be to increase efficacy of the vector transduction and, of course, to anticipate and control transaminitis. A study that I'll give a little more detail on is one through our Freeline spin-out company, which uses a synthetic AAV capsid, and that has a greater efficacy at transducing human as opposed to mouse hepatocytes. The use of a liver-specific promoter and an optimized intron together with codon optimization and the Padua gain of function give one hope of attaining a higher activity at a given vector genome dose. I'm going to illustrate the responses in this particular trial, which have been produced at various meetings. And the first two patients we dosed at a low dose, as we considered it at that point, are shown here. The patients were given prophylactic prednisolone between days 43 and 84. They didn't get any elevation of transaminases at this vector genome dose, and they ended up with factor IX levels in the high mild range between 30 and 50 units per deciliter. Neither patient has had any spontaneous bleeding or any bleeding after trauma under their observation out to just under 3 years now. So if one is, let's say, modest in one's aims and goes for a low dose with a minor immune suppression, one can obtain a very valuable amelioration of bleeding tendency, but it's not in the normal range. So should the patient have severe trauma or require surgery, they're going to need supplementation. Another example from this trial is patient 5, who received a step up in his dose level, but we used a bit more immunosuppression in this patient we, apart from the prednisolone, we used tacrolimus, and we also gave a dose of prednisolone early on when there was what appeared to us to be insufficient control of the elevation of ALT by prednisolone and tacrolimus alone. The patient has eventually attained a steady state factor 9 level around 60% in the normal range, and of course has no bleeding and no therapy. A study I referred to earlier, the SPARK study, uses the Padua variant. The exact details of the capsid vector are not in the public domain, but notable is the fact that most patients attained a factor 9 level high in the mild range. A couple are in the normal range, and one patient has a particularly high level, but another patient is quite low in the mild range. This does demonstrate a approximately 10-fold variation in level, a feature of many studies with gene transfer in hemophilia. I now turn from hemophilia B to hemophilia A. Many will know of the BioMarin study. BMN 270 uses an AAV vector and a construct which Nathwani Research Group developed. Due to packaging constraints in AAV, the B domain of factor VIII is removed and replaced with a short linker, in this case the one that we're familiar with from Refacto, one of the widely used replacement therapies. This has resulted in impressive early elevations of factor IX well into the normal range, or in a few patients somewhat above. But somewhat disappointingly, over time, the level has tended to settle downward. And it's not yet clear exactly where or to what extent the level will fall or continue to fall or settle out. And we're waiting now for phase 3 data from this study out to 4 years to see where this is heading. So I think we need to focus back now on AAV-mediated transaminitis. The first report I've already referred to in the 2006 study, Mano and colleagues, where a subject had attained a level of factor 9. This is not a enhanced activity vector, factor 9, wild type, of around 12%, with a sharp rise in ALT coincident with a fall. Now, this was speculated and subsequently, I think it's fair to say, proved to be due to a T-cell mediated attack on hepatocytes displaying vector peptide on the surface of the transduced cells, leading to a cytolytic attack from T-cell clones, which were either stimulated from memory or de novo developed. In the case of an AAV2 vector, quite likely these were from memory, given that we all get infected with AAV2 in early life. And this mechanism is illustrated on the right-hand side. Our observation in subject 5 I've already referred to, who had a sharp rise in transaminases, and this was coincident with a rise in detectable ELISpot assay response to a pool of vector peptides corresponding to the peptides of the AAV8 capsid. A similar response was observable in subject 6, but a somewhat lower response, and he's ended up, as I've said, with a higher factor 9 level steady state. So potential mechanisms for transaminitis are quite wide when one dials into this more deeply. Now, I've just referred to an early and easily eliminated response, which has good reason to believe is due to AAV capsid. But subsequent studies using higher doses, such as the BioMarin and our own study have resolved that at a higher genome dose, there are effects that are most likely due to features of the AAV genome introduced itself, that is the expression cassette, especially if it contains excess numbers of CpG, which are known to stimulate the TLR9 receptor. And this is an evolved response to recognize foreign viral type DNA. It might be that cellular cess is also involved or vector contaminants. But whatever these mechanisms are, and they're not well studied yet because they're not easily observable in animal models, and indeed we didn't observe them at all in mice, dogs and monkeys before we started the human studies. So we've had to use the human clinical trials to work this out. My conclusion at this point is to point out really that dose response is highly unpredictable, that immune response is also unpredictable, that several mechanisms are involved, that they can cause loss of expression over extended time, and that we may need to redesign our vectors to avoid this phenomenon at the higher vector doses we're now using with the aim of achieving a cure.
Thank you, Ted. And thank you to both our panelists for this particular session. I think we have a lot more time for the question and answers. Can I take the first question? It is to Jay, Dr. Rao. Can we use exosome-associated AAV vectors targeting hepatocytes to increase transgene expression?
Thank you, Dr. Savita. I think the answer is yes. There has been studies where they have used AAVs associated with exosomes, particularly of two serotypes, AAV5 and AAV8, which was also done by Dr. Federico Mingozzi, who is going to be speaking later today. And their group had shown that, in fact, it is highly efficient in delivering the factor 9 transgene into the liver. And they also showed that it is extremely useful even in a setting where there are low level of neutralizing antibodies. So, the answer is yes.
Thanks. Ted, I know you're here, but you're muted. Can I just ask, one of the participants has asked the question is, how do we deal with AAV antibodies present in you and me? What are the possible pre-treatment for these antibodies?
Thank you, Savita, and hello, everyone. Can you hear me?
Yes, we can.
Good. Yes, this is certainly a key question for the rollout of gene therapy in different populations. With the vectors, the serotypes that I've been using, 8 and 5, the local presence of antibodies sufficient to prevent transduction, inhibitory antibodies, runs at around between 30 and 50%, can be higher in some populations that we've studied, which obviously is quite disappointing when you find someone who wants to take part and otherwise meets our criteria. So we've been approaching this along two lines. One is an approach that has been used for reducing antibodies for transplant, quite widely used in renal transplant, is plasmapheresis. And we were able to study samples from patients who had plasmapheresis to reduce ABO antibodies prior to kidney transplant, and study those samples that were collected for the purpose of the transplant. We could look at the rate at which antibodies to AAV8 and 5 were reduced. And we could find that provided that starting titer was not excessively high, say in the range of up to titers, say 500 to 1,000, we could over about see the reduction over 5 cycles of plasmapheresis to levels where we would expect transduction to be successful. So although that's quite demanding, it's very well established technology, plasmapheresis, and we see that as having potential. We've yet to take it into clinical trial. Another approach, another approach that's being pursued is with enzymes that can in vivo eliminate immunoglobulins. A bit more challenging in the case that one is definitely severely reducing all immunoglobulins for a period, but that is in trial in the transplant world.
Thank you. And obviously the holy grail will be when we can redose patients. I have a question for you, Dr. Rao. And although AAV viruses are non-pathogenic and have been reported, but they have been reported to cause DNA damage. So is AAV genome burden in the liver akin to chronic hepatitis C or hepatitis B? Dr.
Yeah,
Rao?
I think that's an excellent question, and I think yes, because we see some manifestation of that in terms of the immune response, at least with the initial studies that we had. Although AAV vectors per se were non-immunogenic, particularly when you test them in preclinical models, the first few trials that was either muscle-directed or even in terms of hepatocyte-directed showed evidence of a CD8-mediated T-cell response. And that is where we had prednisolone and corticosteroids, a transient immune suppression help. But in terms of chronic genotoxic stress, so far, I think, we have patients who have been treated for more than 10 years now. And the evidence shows that there should be certain level of genotoxic stress, but it has not really manifested as an outward symptom as such. And there have been multiple number of preclinical studies with extremely long follow-ups that indeed, even if it happens, it is happening at a level that is not typically detectable or overtly has any phenotypic manifestation.
Thank you. Ted, there's a question for you. From the past experiences in clinical trials of hemophilia B, could you share your opinion on how much factor IX levels Asian countries or LMICs should aim for?
Well, thank you to the question. This has been occupying our thoughts, and I've been discussing this we've been discussing this with Savita over a number of years now. Our experience with the original UCL St. Jude trial, where we achieved levels in the 5, 6, 7, 8% range, was that this completely eliminated spontaneous bleeding in the 6 subjects who attained those levels, but they did get occasional traumatic bleeds. And this did include some sporting, but these were patients who all of them actually had good joint status to begin with. We had a couple of patients at a slightly lower dosing in that original study who achieved levels around 4%, and they tended to get recurrent bleeding. So one needs to, that 5% mark is a good marker for the distinction between moderate and mild in gene transfer. So if one had lower one's expectation with a enhanced activity vector, the Padua mutants, which in effect is what the Unicure study has done. It's taken our exact original, they licensed it from us and put Padua in. And with modest levels, they're getting consistent achievements to the sort of mid-mild to low normal range. And that's very effective. And in terms of ABR going well to 0, I think our patients will teach us just how much of a knock or a blow you can take with a level of 30%. But at that sort of dosage level that we were using, minimal immune suppression is needed. Sometimes no immune suppression is needed. And that in my opinion is a very good result. And patients we've had now followed out to 10 years consistently have a ABR 0. And I'd say that if you want to avoid the risks that we are seeing with the higher doses, the greater immune suppression, in some cases extended loss of activity later on, that a more modest target is acceptable both in low and middle income countries, and as a matter of fact, it should be on the table in high income countries until we learn more about how to prevent the long-term most probably gene-mediated effects that Rao was just talking about. So I put the case for a modest dosing with reduced or no immune response and levels in the mid mild to low no normal as being a very attractive proposition.
Thank you. This is a question for Dr. Rao. What is the scope of using AAV-based therapies for advanced stages of hepatocellular carcinoma and other cancers?
Yeah, I think when we say advanced, if you go according to the BCLC scoring, and if it is stage 3, and if there is some multifocal lesions, then you could still use AAV-based gene therapies to target them. But then, appropriate studies need to be done to see whether it probably completely eradicates this at the advanced stages. So far, no study has been done using AAV vectors to target advanced stage disease. That is number 1. In terms of other cancers, particularly in terms of liquid cancers, yes, there is a scope of doing so, but you need to develop those AAV vectors which have a targeting ability to specifically target those cells that are cancerous. So if you actually equip an AAV vector with the targeting ability, then it is possible in terms of liquid cancers.
Thank you. Ted, there's a question for you. Are there any advancements made towards developing preclinical model organisms to study transaminitis?
Yes, a good question. Indeed, I'm impressed by a fairly recent study from the Jim Wilson group, where they were looking at the effects of the involvement of the innate immune system, where they had given a dose of a vector which was eliciting detectable liver damage in mice. And the study did show that it was TLR9 dependent. Now, toll-like receptor 9 is a receptor that picks up foreign DNA, particularly viral DNA, and is sensitive to CpG and other sequences. And it's a convincing study, and I think that can guide us towards models whereby we could run tests for our future expression cassette, i.e. the specific therapeutic DNA inserts, to pick up whether there's a risk potential for an innate immune TLR mediated response that of course could be chronic, could be involved in the slow late decline of expression levels in, for example, the BioMarin study. So I think it's very important that we look carefully at developing such models. It may be that we can take them even to the level of cell culture models using human hepatocyte cell lines. So I think there's great scope for that, which is certainly to be desired as a preliminary and in a way alternative to finding these things out in the clinic.
Thanks, Ted. And Dr. Rao, a question for you. Given that exosomes appear very attractive therapeutic vehicles with several advantages, do you anticipate a higher risk of adverse events due to enhanced expression of the gene?
Well, enhanced expression of the gene means enhanced expression of the protein, where the exosome-associated AV carries. So in terms of protein expression, it needs to be higher. And it will be certainly higher. And therefore, I don't see a problem with it. But there is a problem which needs to be sorted with the use of exosomes prior to its scale up to humans. That is, right now, because exosomes are derived from human cellular contents or the vesicles that bud out of human cells, the immunogenic property of these exosomes or the membrane proteins here has not been fully deciphered. And this is one aspect that needs to be carefully studied, particularly when we are going to use high-dose exosome-associated AAVs for human applications.
Thank you. I think this is a question from a patient, Ted. As we have seen in the clinical trial data, the factor 8 or 9 level is going down. What after gene therapy, when the patient's clotting factor goes down, I presume it is about whether we can give gene therapy again or not, but I suppose, I mean, certainly factor 8 and 9 will still continue to work. There's no question about that. But is there any possibility of redosing or perhaps not using AAV or another mutant? Is there a possibility?
Yes, we are looking at that. We've, in our original UCL St. Jude trial, where we are out to 10 years now, we've been following AAV8 antibody levels, and they do show an eventual decline to a point where redosing is worth considering, and we're planning to introduce that as a further arm of that trial, with the intention that the redosing would then, these are patients whose levels actually have stayed steady, by the way, in the 5 to 8% range, but redosing to boost them up above in that level. There was a couple of patients at lower dose whose achieved level was 2 to 4%, who would also much benefit because those levels are steady, by the way, would it benefit by boosting them higher into the mild or even normal range. So there are possibilities for that. It might take plasmapheresis or one of the other media, or switching to another vector, but cross-reactivity tends to be quite high. So yes, there is such a possibility. Changing over to use, as we've just heard from Rao, of exosomes would be a way around this too. And we are looking at that at the experimental preclinical level as well. So there certainly are possibilities. And there's going to be quite a lot of patients in, for example, the BioMarin phase 3, I think it's 100, you'd know the numbers better than me, Savita. Is it 120 odd, who in 5 years' time could well be people who we should be looking to offer them a second dose by some means. And progress is great here. With a lot of money's come into the field to push things forward. So if you've had a dose and it's not holding up, don't lose hope. And of course, you're still capable of using any other modality.
Thank you. Again, to Jayendran, there's a question for you. Have you ever tried developing a library of AV capsid mutants and perform screening rather than doing site-directed mutagenesis?
Yeah, I think it's a great question. see, the idea is we have been always trying to focus the basic biology of virus-host interactions, and then try to tinker with those sites that we consider are rate limiting, and therefore the targeted or site-directed mutagenesis approach. We did in one of our earlier times, we did capsid shuffling by a random technique. And then however, our experience in terms of typable or phenotypically useful mutants from that exercise, and also putting them under evolutionary pressure in the presence of neutralizing antibodies, was done during that time. We were not really getting multiple hits that may have been useful for us. And then we completely switched over to doing, virus host relationship, their understanding biology and site-directed mutagenesis. Having said that, it was our experience, but there are groups like Dr. David Sheffer's group at the UC Berkeley has pioneered capsid shuffling and evolutionary mutagenesis of AAV vectors, and they have come up with several AAV vectors that shows enhanced delivery and lack of immunogenicity in multiple model systems. So yes, it is doable, but in our case, the experience was not so good. So we kind of stuck to more site-directed and biology-based approach.
Thanks, Jendran. I think we are coming to a final couple of questions for Ted. Ted, there are two questions, and I'll ask both of them at the same time. What age can we actually start with gene therapy? And secondly, your data for the last 4 year, for the 4-year data is very good. After what time do you reach a steady state? And can functional studies of T-cell subsets predict inhibitors and transaminitis?
Okay, so I'll take the first one, and that's the age. We've currently started at 18, but we have plans to move next into the teen years, down to the 16, then 12 to 16. As we move down towards the pediatric under 12 level, we've got to bear in mind that there may be need for redosing because the liver won't be at full size. The liver is near full size by 12. So we don't anticipate fall-off problems treating anywhere down to the bottom of the teen range. But once we go into the childhood, we'd like to be able to offer a redosing potential from that stage. Pediatricians are naturally, and I understand this, cautious, so they're not ready for us to go pediatric yet with hemophilia A and B. We are in development of a factor 7 vector, and their early treatment is definitely where we're going with that vector. So we probably get some experience in early childhood in the rare cases of severe factor 7 deficiency. Okay, I hope that's covered that for you.
Thank you.
Now the second question was about T-cell types.
Yep, and to predict transaminitis or inhibitors.
Yeah. Well, we've of course selected patients for the trials who are up to now, we've been choosing patients who've never had inhibitors and who are very well tolerant of wild type factor 7 or factor 8. But in the factor 8 region, we're about to start. We are in fact recruiting to a trial for inhibitor neutralization by gene transfer using the biomarin vector initially. So we are going there. We're also planning to extend that to subjects who've had a transient inhibitor in the past as a further step, because they have been excluded up to now. So we are broadening our remit to include patients who've had or currently have inhibitors. T-cell subsets are oh, and by the way, in terms of inhibitor, what we're doing is essentially immune tolerance by endogenous production, a vector which David Lillicrap has shown to be effective in his dog models of factor VIII inhibitors. For the T cell subsets, this is crucial, because one of the things we've learned more recently in our studies in a GO8 trial, which hasn't yet been updated much in the literature, but is producing some stable long-term maintenance of factor VIII levels in the high, mild, and normal range now. We've learned, and from that and from the freelance study I presented some data from, that it's crucial at higher dose levels to be able to allow tolerance to develop. And that means Tregs come into play. And they're the tolerance generating T cells, which can suppress the innate and acquired immunity. So it's the balancing of the other side. So we are looking at T cell subsets in our studies going forward.
Thank you. There are a number of questions, and I will ask if the ASGCT team can answer those questions on the question and answer panel, and we probably could send out an email around. Thank you very much for the first session. And we will have a break for the next 15 minutes, and we restart in Indian time at the time that we have said in 15 minutes. Thank you. Thank you all.
Thank you. Thank you very much.
Thank you.
Welcome back to the next session. This is on CAR T-cell therapy, opportunities and challenges. The first speaker for this particular session is Dr. Geeta Jotwani. Dr. Jotwani works as the Deputy Director General at the Indian Council of Medical Research. She's been responsible for the national guidance for gene and cell therapies in India and is also a member of the WHO Cell and Gene Therapy Group. She's going to tell us about the guidelines in India and educate us a bit more on how to go about with gene and cell therapy clinical trials. Thank you, Dr. Jotwani.
Thank you, Savita, and thank you, ASGCT, for this opportunity. I'll share my screen. Can you see my slides? Shall I go ahead?
We can. Yes, we can.
Yeah. Good afternoon, everyone. And I'll be speaking on the national guidelines for stem cell research and gene therapy. So as you all are aware that this is the era of new generation therapeutics products for different modalities like stem cell and stem cell based products. Then we have gene therapy products and we have immunotherapy products through different modalities. It is not like a one conventional way of therapeutics. So, and this is quite challenging area. And with the developments in the field, it is very important to guide scientists and the clinicians working in the field. So in India, the scenario was like, I'm talking about 20 years back, there was a Drug and Cosmetic Act 1940 and rules 1945. There are several amendments of this in a due course, and it is a continuous evolving area. So these have been updated periodically. Then we had Indian Medical Council regulations, which are for the professionals to conduct etiquettes and ethics in the biomedical profession. And then we have a Drug and Magical Remedy Act. This is just related to cell and gene therapy. So I'm quoting these acts, which are quite old acts. But very recently, we have the new drugs and clinical trial rules 2019, which covers all these new generation therapeutics. So I'll talk about this later, about the new drugs and clinical trial rules in context of the guidelines. But first, I'll make you all aware about what are guidelines and how we went about it. So this is our journey of ICMR, ICMR being the apex advisory and technical body under the Ministry of Health and Family Welfare of India. We have been conducting, as per the development in technologies, we always are proactive in conducting brainstorming sessions to deliberate upon the issues and concerns and ethical and scientific issues related to the technology. So we have been very proactive, and way back in 2001, we, when the embryonic stem cell research hit the news, we conducted a brainstorming, and our journey started from there. And we were requested by our ministry to develop guidelines for stem cell research and therapy. And that is how in 2002, because the focus was more on the embryonic stem cell, so we developed this draft guidelines for stem cell research and regulations 2002. Followed by that, very comprehensive guidelines were brought in by ICMR in collaboration with Department of Biotechnology, which falls under the Ministry of Science and Technology. That is the guidelines for stem cell research and therapy that happened in 2007. And as the technology developed, it was a continued process of evolving and giving directions to the stakeholders. So we have continuously revising these guidelines. The next happened in 2013, and after that, the 2017 guidelines of stem cell research and therapy. Along the side, ICMR is being involved with development of ethical guidelines for biomedical research and health research involving human participants. And that is also have been continuously revised in 2006 and then 2017. And along the side, with the development in the field and recent development, we thought that we should have guidelines for gene therapy product development and clinical trials. So those were released in 2019 and followed by, as per the request from the ministry, we had to formulate national guidelines for hematopoietic stem cell transplantation. And of course, to curb the unethical practices in the country, it was important to educate the stakeholders about available stem cell therapy, and which is a proven therapy. So we had to put up a document on evidence-based status of stem cell therapy for human diseases. And these both documents we released in January 2021. So for past 20 years, I have been involved and actively working on these documents at the ICMR front. So as I said, the development and the harmonization is the integral part. When we are developing some documents, we have to harmonize it with existing acts and rules, and also to update as per the recent scientific development. So there is a continued interagency, interministerial dialogue and the consultation to review and revise these documents. So the ethical principles have been well defined in our national guidelines for biomedical and health research involving human subjects. So the basis of all these guidelines, the ethical basis of these guidelines is based on this document. So, and this has been legitimized and is part of the new drugs and clinical trial rules. And this very clearly mentions about the stem cell guidelines that whoever is working in the field of stem cell research should follow the national guidelines for stem cell research 2017. Those are the latest guidelines, and we are in process of revising those, and that will continue. So the basic ethical and scientific considerations for both stem cell and gene therapy products guidelines are defined here. So basically, the most emphasis is on the safety of donors, then the manufacturing and quality assurance, of course, to curb the commercialization of unapproved therapy, and promoting evidence-based application of these new generation therapeutics. So in case of gene therapy products, as you heard in the previous session, that you know that these kind of a new generation technology, which has a very, they have multiple components of gene therapy, and there are different modalities of gene therapy. To achieve that successful gene therapy corrections and protein expression, it is important to have an appropriate selection of gene therapy vectors and the gene of interest to ensure clinically relevant expression and correction and for the particular disease or condition. So preventing unwanted side effects and unnatural advantage. So putting those scientific and ethical consideration in mind, we have framed these guidelines. So coming to our national guidelines for stem cell research 2017, these are the contents of the guidelines. I'll not go in detail, but it covers almost all the components what our expert committee and the drafting committee thought of communicating or disseminating to the masses. So these are what kinds of stem cell research is covered by these guidelines. It actually guides for all both basic and translational research. The stem cell research involving human stem cells and their derivatives, but does not apply to non-human stem cell. And the approved therapies like hematopoietic stem cell transplantation, which is approved in hematological cancers and some disorders, which has been approved by and listed at American Society for Bone Marrow Transplantation and European Society for Bone Marrow Transplantation. On the basis of that, we have also annexed the list of approved indications to educate the masses that these are the only approved indications of stem cell, not the others. So, and which are those who are under development have been clearly emphasized. So, who follows the national guidelines for stem cell research? These are for all the stakeholders. For some stakeholders, it is for education, but most of the stakeholders like researchers, organizations, sponsors, industry, and of course the oversight regulatory committees. So this is a guidance for all the stakeholders. It is very educative, and this has made real impact in past few years. There is a monitoring mechanism suggested under these guidelines at the national level and at the institutional level, just to inculcate ethical practices and scientific practices in the amongst the scientists and clinicians. This kind of mechanism has been built, and the National Apex Committee for Stem Cell Research and Therapy is where the secretariat is with ICMR, and I'm the member secretary of the same. And we registered all these institutional committee with National Apex Committee. So, and we monitor their research activities closely to look at whether some people are involved and following and working in compliance with these guidelines. So these guidelines are basically the research categorized in these guidelines based on the source of stem cells and level of manipulation. So the research activities have been categorized under permissible, restrictive, and prohibited areas of research. Permissible and restricted are basically permissible areas of research with extra oversight or the approval of the committees. And prohibited is, these are all made in very much in harmonization with the international regulations. So particularly 2017 guidelines, we definitely went through all US FDA and EU, as well as TGA and even Japan regulations to update these guidelines. The level of manipulation, based on the level of manipulation, the research has been categorized into minimal manipulation, substantial or more than minimal manipulation, and major manipulation. So under the minimal manipulation, there is only, which is defined as processing neither alters the number nor the biological characteristics and the function of the cells or tissue. So, whereas substantial or more than minimal manipulation involves ex vivo alteration in cell population, which is not expected to result in alteration of cell characteristics and function. And whereas major manipulation, where the genetic and epigenetic modification leading to alteration in biological characteristics and function is involved. So based on these two aspects, the research has been divided into permissible area of research, which is basically a basic research approved by at the institutional level to inculcate ethical and scientific practices by Institutional Committee for Stem Cell Research and Institutional Ethics Committee, which is generally known as IRB. And then clinical trials it's all permissible only with as defined in these guidelines, that all the clinical trials, the cells used for these clinical trials has to be a clinical grade cells and to be processed and certified by facility, certified by CDSU, processed under the GMP facility certified by CDSU, and needs the approval at institutional level as well as at by a committee established by Central Drug Standard Controlling Organization, which is the Indian FDA. So without their approval, they cannot conduct any clinical trial. So under the restricted area, as I said, that we need an additional approval as per the area of research. So for the research involving introduction of human embryonic or iPSC or somatic cells into animals, they need, and also the studies on cameras including primates, they need the extra approval, and that they need the approval of National Apex Committee for Stem Cell Research and Therapy. And as well as when you're doing animal studies, you need the animal ethics approval and approval of CPC-SCA, which is an organization who controls the animal ethical studies using animal models. When you are involved in genome modification, including gene editing, there is a review committee for gene modification who reviews the applications involving gene modification. All application at preclinical stage are enrouted through this committee. And finally, once the approval of this committee is obtained, then one can enter into the clinical phase, and after obtaining a regulatory approvals from the Indian FDA, that is CDC SCO. For the international collaborations, there is a separate committee for international collaborations, which is a health ministry screening committee, which has been, for which the secretariat is ICMR, and the Director General ICMR and the Secretary Department of Health Research is the chairman of that committee. So all international collaborations are approved by this committee at the final stage. Just to list few, whatever prohibited areas as per the national guidelines 2017, that human germline gene therapy and reproductive cloning is prohibited, in vitro culture of intact human embryos, clinical trials involving transfer of genogenic cells in human hosts, then clinical research on genogenic human hybrids, use of genome modified human embryos, germline stem cell or gametes for developmental propagation, and implantation of human embryos after in vitro manipulation at any stage of development in the uterus in human or primates, and the breeding of animals in which any type of human stem cells have been introduced is prohibited as per these guidelines, and which are very much in line with the international regulations and in harmonization with the guidelines. So the stem cell guidelines, we have also given the additional directions and informations for the stakeholders, defining about the composition of institutional committee for stem cell research, and what is the requirement for registration, and what is the composition of National Apex Committee, and how it functions, and what are their term of references. Then, of course, the template for clinical trial protocol. As I was saying that we have listed this in the annexure, the approved indications of HSCT on based of EU BMT and ASBMT. Then screening on screening of donors, the requirements and the format for allogenic transplantation, then manufacturing of stem cells and their derivatives, and release criteria for stem cell. This is just to substantiate existing regulations, because there was a big gap from Drug and Cosmetic Act till formulation of NDCTR, that is New Drugs and Clinical Trial Rules in 2019. So these guidelines were just to substantiate and direct clinicians, industry, and other stakeholders that what regulatory rules they have to take, and to help them and handhold them for the regulatory approvals. So the main challenges we faced with the stem cell guidelines was the minimal manipulation, and this is a global problem. It is not only in India. We realize that we have been facing this everywhere in the world, that many people are practicing minimal manipulated stem cell therapy for non-homologous use, or for the indications which are not approved. And that's why we had to really struggle to frame this definition, and we are still having debate on the same. There was always been communicated to our regulators that we need to have framed these new drugs and clinical trial rules to substantiate these guidelines, because many desperate, vulnerable population was falling prey to these unethical practices. There was a financial toxicity because these being charged unapproved therapies. Then internationally, we have been criticized There was no systematic study done by these folks to understand safety and efficacy, what they were offering. They were false promise or hope to cure incurable diseases and conditions, and several complaints received by government organizations, including ICMR, from patients, NGOs, and many patient advocacy groups. So keeping minimal manipulation outside the purview of the definition of new drugs was a real concern for ICMR and many scientific community, that this should be put under the purview of new drugs and clinical trial rules. And there were continued discussions and debates happened. So I'll tell you the outcome of that at the end of my presentation. So coming to the national guidelines for gene therapy product development clinical trial, which were released in November 2019, these were particularly developed for in the same direction to guide the stakeholders that how should they go about gene therapy product development, realizing that many of the stakeholders were involved in developing these products, and they were at the early stage of product development, so it was appropriate to guide them. So I'll skip this slide. Okay, so the contents of the guidelines. So as the framework of our guidelines are more or less similar, except the technical part of it is different. So here are the guidelines for gene therapy product development and clinical trials, which have been notified by ICMR along with Department of Biotechnology and Central Drug Control Organization. So they are the signatory on this document, and this is based on the general ethical principles, then specific scientific and ethical considerations. We have classified gene therapy in this. We know that there are several different modalities of gene therapy, so it was little challenging for us to frame this. These guidelines are just based on the conventional gene therapy, but we are now working on developing a process and disease specific guidelines. So which also has defines the mechanism for review and oversight. I'll describe that in my slides later on. The responsibilities of investigators, institute sponsors, and ethics committee and other concerned committees. Consideration for CMC and quality control, assurance and product attributes for human gene therapy products. Then definitely the requirements for GMP facility, infrastructure, and personnel. The requirements for preclinical evaluation of these gene therapy products and clinical trials. And what are the requirements for international collaboration or procurement of GTPs or vectors or the components of GTPs, import and export of those. Then, of course, the awareness and education of stakeholder is a very important part of our guidelines. And publicity and advertisement in all media, including electronics and print media. So we are guiding that through this. And periodic updation, which is an integral part of any guidelines. So this is a schematic representation of designing a gene therapy trial application, though it's like the details are there in the guidelines. Those who want to refer to these can find and can also put the suggestions if they find any gaps, because it's a continued process. We'll be updating it periodically. So there are different stages of gene therapy development. So right from selection of gene of interest, first the disease and then the gene of interest, then the vehicle for gene delivery, and looking for a preclinical model to study its safety, and then of course further testing these gene therapy products for different route of administration and dosages. So this next stage for gene therapy production, product production, that for the relevant disease selection of GTPs, CMC for these GTPs, which is important, and then GMP production for the clinical grade gene therapy products, and testing for its integrity and then packaging and storage. So this has been well defined in this document. And of course, the preclinical testing, which I already mentioned, some of the components of preclinical testing, that is selection of relevant testing models, which is been under discussion. There are a lot of questions from the stakeholders that what are the models, because for most of these diseases, there are no relevant models are available. So studying the toxicity of the product developed in a GMP facility, then its biodistribution, then gene transfer efficacy, therapeutic benefits, and biomarker testing. And the next stage is for the clinical study design. So moving on to the next slide, I'll tell you what are the requirements for clinical trials, the study population, appropriate looking for a history of the disease, and patient selection criteria, followed by study design, consideration for investigational new product and imported product, whichever has been selected for the kind of clinical trial ones want to conduct. The study design with dose selection, safety considerations, efficacy considerations, follow-up time periods and the long-term follow-up has been very clearly defined in this document. So of course, there has to be a follow-up with the patient's pre and post experience after obtaining gene therapy. And we were recently discussing about having a separate informed consent for the patients involved in gene therapy clinical trials. So, and of course, the personnel involved in the gene therapy clinical trials, their expertise, their qualification has been well defined in this document. So this is a regulatory approval process as per present scenario in the country, that whoever is interested in gene therapy product development, they are going to put up a proposal. They are first going step by step in the production of these modalities. They are going to have a biosafety committee with the institute involved in gene therapy product development. At the preclinical level, you require the institutional biosafety committee, their approval, their registration with RCGM, which is a review committee for gene modification. And then for preclinical studies, the approval of RCGM is important. And once at this stage, once you get through the preclinical stage, then it comes the ethics committee involvement for approving the clinical trial proposal, which is then routed to the regulators, that is CDSCO, via Gene Therapy Advisory and Evaluation Committee. The secretariat for the same is again at ICMR. And once you start the gene therapy, once you get the approval from regulatory approval, then only you can start the intervention and recruit patients. And before that, you have to take the approval of ethics committee before you start clinical trial. So this is the regulatory path for gene therapy product approval. So as I mentioned, there is a committee, the mandate of this committee, which has a secretariat at Indian Council of Medical Research. This committee is mainly been established to provide handholding and free clinical IND consultation for investigators and industry, and robust evaluation of GTPs and gene therapy clinical trials, providing technical and scientific acumen to our regulators. Then formulation and updation of these guidelines and education and awareness of stakeholders. The mission of this committee, particularly national committee, is to streamline regulatory framework in coordination with various government agencies, scientific and ethical conduct of research and development of good and clinical grade gene therapy products. Safety of human participants is utmost important, and at the same time facilitate indigenous development and affordable gene therapy solutions to Indian patients. So this is the website of the gene therapy, which is recently developed, and we are going to have an online submission of gene therapy applications before going into clinical trial stage, and will be evaluated by the multidisciplinary committee established by Department of Health Research, having expertise in the field. So coming to the regulatory discussions on the new drugs, because as per the existing regulations, stem cell and gene therapy products have been given a definition of new drugs. So yes, stem cell and gene therapy products, even immunotherapy products, they fall under the category of new drugs. So what is the strength of this regulatory framework? We awaited, I mean, this was a very long awaiting step by the Health Ministry, and this document, which was then the rules which were released on 19th March 2019. So it's giving a good clarity to the stakeholders about what falls under the category of new drugs, because there was a big gap under the Drug and Cosmetic Act. Even after the amendments of our old act, there was a big gap defining what are the what is the regulatory path for stem cell and cell-derived products. So it very clearly defines that these modalities, these new generation technologies will always remain as new drug. So no academic clinical trials for knowledge enhancement can be done using these new generation therapeutics. At the same time, the guidelines were developed by ICMR, they substantiate further elaboration about all the requirements for these application and clinical trials as I defined in my presentation. So moving on to this, the only consideration which the scientific concern which scientific and clinicians had was this new drugs and clinical trial rules does not elaborate on what constitutes stem cell derived product and GTP. And it's very ambiguous about the definition of the level of manipulation. So we have been debating, and very recently they have released a document that minimal manipulation does not fall under the purview of product. So that has been put outside the purview of, and which is still challenging that how we can curb unethical practices in the country. So these are the directions issued recently by the Health Ministry, clarifying that how they have explained that why and how minimal manipulation does not fall and cannot be defined as a new drug. So there are contentions that minimally manipulated stem cell products, that derived products, means a drug which has been derived from a processed stem cell and which has been processed by means of only those which are processed by means of substantial or more than minimal manipulation can be covered under the definition of stem cell or cell-derived products. Whereas minimal manipulation, cells removed from an individual for implantation of such cells only into the same individual for use during the same surgical procedure, should not undergo processing steps beyond rinsing, cleansing, or sizing, and these shall not be considered as processing, and these will be defined as a minimal manipulated cell, and they do not fall under the purview of drugs and this new drugs and clinical trial rules. So it's still under debate, but this is defined for the understanding of the stakeholders. So cell-based products and tissue-based products which have been processed by means of substantial or more than minimal manipulation are covered under the new drugs and clinical trial rules released in 2019. The specific considerations after these guidelines, after looking at these regulations and guidelines that there is a structured multi-layer monitoring involving institutional biosafety committee, review committee for gene modification, and the regulator, that is CDSCO, and routed through GTAEC, is providing a very robust mechanism for approval of stem cell or gene therapy based products. So, and stem cell products, there is a separate mechanism where it is an institutional committee, institutional ethics committee, and then there is a regulatory committee. And in case of some concern like embryonic stem cell and iPSCs, it should be routed to National Apex Committee for Stem Cell Research and Therapy. And rest of the prohibited areas, which have been very well elaborated in these guidelines. The germline or in utero gene therapy is prohibited in India. Somatic cell gene editing is permissible, but needs to assure no biodistribution to the gonads. Therapeutic use of stem cell for unapproved indication is not permissible, only to be done under the realm of clinical trials. Investigators involved in clinical trials with CGTP must have domain expertise and GCP training. So infrastructure has to be GMP certified by the regulators, that is CDSCO, and the challenges are always there, and it's global challenge to curb unethical use of stem cell and cell-based products with escaping the regulatory pathway. As I said, that ICMR has been involved in awareness and education to the stakeholders, and we have been conducting dissemination programs. We have conducted public consultations and taken a view of stakeholders, and the revision of these guidelines is the inbuilt part and will be continued process as there are developments in scientific developments as well as some regulatory updates. Thank you. This is the schema of our clinical trial approval for gene therapy and stem cell products as defined in my presentation. So,
thank you, Dr.
Thank
Geeta. I think that was an excellent overview of the regulatory landscape for both cell therapy and gene therapy in India. There are several questions as usual. We will take it at the end during the panel discussion. So it's my great pleasure and privilege to introduce my next speaker, which is Dr. Vikram Matthews. He is currently a professor from the Department of Hematology at the Christian Medical College in Vellore in India. He has extensive experience on various aspects of diagnosis and treatment of hematological malignancies, and credited with multiple cost-effective innovative solutions for the treatment of hematological cancers. And today he will discuss on the current Indian scenario with CAR T-cell therapy. Dr. Vikram.
Thank you, Dr. Jayendran. I'd like to thank Dr. Savita Rangarajan, Dr. Jayendran, and ASGCT for this opportunity to present at this exciting forum. That was a great talk by Dr. Geeta Jotwani before this. It spells out the challenges that we have and also the need for caution as we proceed with any of these therapies. So I'll be talking in the next 20-25 minutes on CAR T-cell therapy, the Indian scenario. And the broad objective is to take you through the current status and challenges with the existing models. It's a fairly focused audience, so most of you all are familiar with many of these things. I will not be very data intensive in this presentation, but the objective is to give an overview. I'll talk about some of the industry activities that I'm aware of and some of the academic centers. I probably will not do justice to this area because I will not know every single industry and every single academic center that has a project going on. But what is there in the public domain or who have communicated and people have responded is what I will be addressing, but I'll give you a sense and a flair of what is happening in the country. I'll try to talk about the need for local regional programs, both in terms of vector development and cell manufacturing. And I'll talk a little bit about our experience with the regulatory process, which Dr. Geeta has covered exhaustively, and some possible strategies to improve access and goal for equitable healthcare distribution, which is what we all try to work for. A brief introduction for students in the audience. Basically, CAR T cells are using a conventional component of an antibody which recognizes the specific antigen. And to this, you put in elements by which, if you introduce it into a cell, once the antibody component interacts with the antigen, it does not require all the other elements of the CAR T cell, such as the co-stimulatory molecules and additional recognition sites by the MHC receptor, etc. It has all those domains within its structure, the activating co-stimulatory domains and the activation CD3 zeta elements, which can activate the T cell. So while the monoclonal antibody component gives the specificity, but antibodies are not as effective as a T cell in bringing out kill. So a CAR T cell essentially combines the high affinity and specificity of the antibody along with a high potential for cytotoxicity and proliferation. And more important, you can guide the T cell to an antigen of interest. So in the case of malignancy, for example, in ALL, where there is a unique antigen called CD19, you can direct the T cells or redirect the T cells to attack these cells and kill them. And that broadly is the principle. There are different generations of CAR T cells. And most of the current CAR T cells are the third or fourth generation CAR T cells. There are two major elements when we talk about CAR T cells. One is the vector production. That is the discovery, development, and large-scale industry-level manufacturing of these vectors to cater to a large population. And the second is using these vectors to produce the transduced T cells that can happen either centrally or, as I would like to present, decentralized at a center where it's going to be used. That this CAR T cells has been a breakthrough and is something that all of us are aware of. It has been one of those therapies that has really changed the face of the outcome of many conditions, as I'd like to point out. And this has led to a very rapid, I would say, in conventional approval stages, when you look at drugs, this is an incredibly rapid pace. Between 2017 and 2021, there's a whole series of molecules that have been approved and are on clinical trials and are also in current guidelines. But the common theme, unfortunately, is that many of them are industry-driven, though many of them had their birth in academic institutions. The spin-off companies that come from them are usually industry-driven. They're centralized manufacturing process. And as was pointed out right in the beginning, the cost of this is prohibitive to a point where it is really not something that's affordable for an average patient or even the exceptional patient in India for that matter. That this is an incredibly effective therapy has been highlighted by, as you can see here, the very first patient to receive a CAR T cell therapy for ALL over the years. In patients who do well with this therapy, they really do well. And this is a group of patients who I will show you in the subsequent slides had an incredibly poor outcome with conventional standard of care. Before we go forward, I think it's important to look at CAR T cells in the context of an allogeneic stem cell transplantation, which is the other way of using or leveraging the immune system to attack malignant cells. I'm kind of focusing on the malignant cell part. In India, MSD stands for matched sibling donor, MUD stands for a matched unrelated donor, and a haplotransplant is usually between a father or mother or a sibling who is not fully HLA identical. And those are the costs. Even these costs, which are about 1/10 or less of what it is in the West, is still unaffordable for the majority of patients. So it costs between 14 lakhs for an MSD, 30 to 40 lakhs for a blood transplant, and 25 lakhs for a haplotransplant. The cost for a Western audience is illustrated over there in US dollars. These costs can double if you have significant complications and go to the ICU. Now, with that standard of care, let's take ALL as an illustrative data from our center of potential of a group of patients who could potentially be a potential target for CAR T cell therapy. If you look at ALL patients who relapse early, when we say early in the context of ALL, it's on therapy or within 2.5 years of starting therapy and relapse refractory, that is the kind of long-term event-free survival you are going to see. That is the kind of standard therapy, which in the earlier slide I showed you, Emily White would have fallen into. The outcomes with therapy is incredibly poor, and many of the malignancies that we are looking at treating fall in this kind of a range. They are relapsed refractory patients who have exhausted all standards of care, and by conventional senses would have gone either on palliation or in the setting of, or would have been enrolled onto a clinical trial. There are very limited options in our country for many of these patients. Some go out of the country who can afford therapy, usually to the Far East or to China. So the ongoing activities in India, I'm going to break it up into industry and academic centers. And I really like to stress very early that this is not an antagonistic relationship. I think that it's very important that there is a symbiotic relationship within academia and industry, provided there's mutual respect and gain for all. Because industry has a very important role to play in scaling up vector production and access, especially in a country of our size and complexity, or for that matter, in any country. No single academic center could possibly have the drive or the wherewithal to actually cater to that. Academic centers, it's very important role in constant technology development and there's a need for robust funding mechanisms and capacity building. When I say constant technology development, it's not just about developing new vectors. It is also about strategies to develop low-cost delivery of these technologies to patients so that access improves. And there are different strategies for that, which is way beyond just developing an indigenous vector. Even developing indigenous strategies at which transduction can be done efficiently at a low cost and also training, capacity building of technicians, clinicians to take care of these patients, which requires special skills, is important for this technology to be easily available and accessible to the majority of our population. When you talk about industry, there are a few people that come to mind immediately. The first among them is Intas Pharmaceuticals, and they have actually invested a substantial amount in a separate section called Cell and Gene Therapy section. This chart over here shows their existing pipelines. They are actually collaborating with Miltenyi Biotech for their CD19 CAR T cell, and they're also working on developing indigenous vectors for the diseases shown over here. Most of them are still in development phase, and they have not yet reached a clinical trial. So that is Intas Pharmaceuticals, and they are collaborating with Tata Medical Center in Kolkata, where they have a facility for their translational work. Immunil, on the other hand, is a company based in Bangalore. I did write to them, they did not get back, but this is basically from their website where they've been working. They've been a lot of attention to the media for the work that they have recently been proposing to do. They are also, from my understanding, in early stage preclinical level of development. And they are working with Narayan Hridayalaya Center in Bangalore, which is their translational center. The other group is Origin, which is a wing of Dr. Reddy's. And they are also into this technology and investing in developing a GMP CAR T cell facility. They have already in-licensed a product, which is one strategy many of these companies are following for many products. And they've already announced their plans to start therapy in relapsed refractory multiple myeloma. When you come to the academics, what has really pioneered in this field is Dr. Rahul Punwar and his group at IIT Mumbai. This has been a spectacular body of work, truly. They have actually developed an indigenous vector, a CD19 vector, which has enough novelty that it has been given a novelty patent. They've actually taken it through all the steps with their clinical partners at TMH Mumbai, Dr. Gaurav Narula, Dr. Hashmukh Jain and their teams. And they have produced this CAR T cell vector, which in their preclinical work have shown to have less cytokine release. And they have infused 5 patients in the recent past, my recent communications, and none of them at this stage, from again, personal communications, have had a significant cytokine release syndrome or neurotoxicity. The next 3 slides are straight from Dr. Rahul Punwar. Grateful to him for providing these to me. It just outlines the timelines that they have taken to get to where they are. So started almost 7 years ago, and they've gone through the whole process of preclinical, developing the vector, truly an admirable body of work to have translated this to the clinical. And as I said, 5 patients have been dosed at this point in time. They collaborate with the Tata Hospital, as I already told you, ImmunoAct, which is a private limited company, which Rahul Punwar works with as well. And the knowledge partner is National Cancer NCI USA. There's been a lot of attention to their work, as is shown over here in a slide shared by Dr. Rahul. And this is good for everybody in the field because it's taken attention to up to the Prime Minister's office. And we hope that this will translate to increased funding, because it's important that attention is drawn to this and funding is distributed and capacity is built across the country to make this therapy accessible to all our patients who deserve this therapy. So coming to the need for a national program and focus, one is technology development. Obviously, it's important for us to be in the front line in terms of technology development. We need a program for indigenous vector development and large-scale manufacturing. This has to be an academia-industry partnership, in my opinion. And we have to look at strategies to decrease cost of cell manufacturing, including what I'll be talking about. What we work on is decentralizing CAR-T cell manufacturing. We can harness the higher national purchasing power parity that exists in our country and the lower labor costs favorably for our patients. We can harness the scale of production and numbers to reduce the cost. The need and the demand is high enough that if you introduce enough players, that is including industry and academic centers, etc., by the competition itself, the cost, I'm fairly confident, will reduce. So coming to the CAR-T cell manufacturing, the conventional model that is followed at present is the cells are shipped from hospital to centers, which could be across a continent or it could be across a country, and where the cells are then transduced and then shipped back to the hospital for the care. And this is usually driven by centralized industry-driven structures where the cost can be very high, as has been shown in the US, but it's possible that these models can be replicated in India at a lower cost. The other model is to decentralize it, where, for example, the vector can be shipped to the hospital, and the manufacturing of the cell per se can happen as a point of care. This is just an illustration of a system using the Miltenyi Biotech. This is in the US. And here you show that it actually sits, the system, which is a closed system, sits in a lab, which is just about a class 100,000 facility. The importance of that is that the cost of maintaining, setting up is one cost, but the cost of running and maintaining a class 1000 facility long-term for semi-automatic open systems is what adds significantly to the cost. So the major challenge has been cost, and this is dictated, as I've kind to illustrate, to a large extent due to industry and centralized manufacturing process. Centralized manufacturing process is also going to be a logistic challenge in our country, and I don't think any academic center can actually produce vectors at a scale that is required for everybody in this country. If we can have a decentralized manufacturing process, what we are using, the Plenimax Prodigy system, is something that we have done preliminary work on. So this work is done in our center. We have a transplant program, which does about 250 transplants a year. Even in the COVID year, we did close to 160 transplants. We've been working with Lentigen and Miltenyi Biotech Company, and we've got an agreement with them to use their vector. That's the details of the vector. I'm not going to go into the details, but it's a conventional third generation anti-CD19 vector, which has already been used effectively in preclinical and early phase clinical trials, both in North America and in Europe. We use our GMP facility to manufacture our T cells. It's a class 10,000 facility with a Prodigy sitting in it. And basically, the entire system is a closed system where the selection of, once the AFRS product is connected to this and the appropriate reagents are connected, the selection of the T cells, the transduction, and the expansion between 9 to 12 days as you decide fit, and the cell comes out, and you have to take it through a release criteria, and the cells are ready to be used. We've done preclinical validation runs. This just shows you some of our data. In our pre-validation runs, the transduction efficiency is consistent and reproducible, very similar to data that has been generated in North America and Europe, varies between 50 to 60%, 66% in these particular runs. And we chose to take the expansion for 9 to 12 days, at which point you can get between 28 to 47-fold expansion. And the majority of the T cells breakup is shown over here, as is the pre-transduction and post-transduction number of CD4, CD8 cells. And there's an enrichment of central memory T cells, which is also a good thing in terms of efficacy, which I'm not going to go into detail, because we anticipate there'll be less cytokine release syndrome with that kind of a T cell profile. All the other aspects in terms of the vector copy number is all within fulfilling criteria. There is no replication competent lentivirus at the end of the product, as more or less expected. With point of care, or it's also used point of care or a decentralized or distributed manufacturing, these are some of the terminologies that are used. there is significant cost reduction because this closed system, the maintenance and running of this closed system is significantly lower. We believe it can be scaled up very rapidly to multiple institutions. There is an initial investment, but the long-term running cost is, we think, very, very low compared to running a semi-open system for manufacturing T cells. In addition to that, these systems you can integrate. So for example, if Rahul Punwal's vector becomes commercially available, we can easily integrate it into the system. And along with regionally developed vectors and reagents, we can further reduce the cost. Currently, we've estimated the cost to be around rupees 25 to 30 lakhs, but we think that we can significantly reduce these costs further. Walking you through the processes that we have gone to get where we are, many of this has been covered by Dr. Geeta, but essentially this has been almost a 2-year exercise. We had our initial IRB, institutional research approval. Then we had to take the institutional biosafety committee approval. We had to go to the review committee on genetic manipulation to get permission to get the vector into the country and do all our pre-validation runs. We've completed that work and submitted a report, a detailed report to them. We also went through the GTAC, which is the advisory committee, which has looked through our proposal. And right now, our proposal for a clinical trial sits with the CDSCO for approval to start our phase 1/2 clinical trial. We also work with other, as a center, we work with other groups in the country. This is a publication looking at Global Gene Therapy Initiative, GGTI. This is a group of individuals from centers from all over the world. Fred Hutchison is, you can see Dr. Poonam Malik there from Cincinnati Children's, etc., along with end user countries, including India, Africa, Europe, and North America, where there's a common goal of developing vectors and strategies to make both all these gene therapy initiatives, initially for sickle cell and thalassemia, but also for CAR T cell therapy, affordable and we hope universally available and accessible to patients. This access to patients is actually not just our challenge, it's also a challenge in the West, where for various reasons, it's not easily accessible to all outside the setting of a clinical trial. We also have an MOU with Origin, which is a branch of Dr. Reddy's, as I already mentioned, and we hope to be working with them on the multiple myeloma project. I'll stop here and thank my collaborators and people who actively work with me in manufacturing and in our T-cell program in our center. So Nedin Cherian from SiteCare, Dr. Boro Dropolik and Milteni Biotech, other staff, students and colleagues, and of course my funding agency, ICMR. Thank you, and I'll be happy to take questions at the end of the session.
Thank you, Vikram. That was an excellent talk. I would now like to go on to introduce Professor Persis Amrolia. Professor Persis Amrolia serves as a professor and a clinician scientist at Great Ormond Street Hospital. He has a long-standing interest in the area of CAR Ts, and Persis will be talking to us about his experiences in running 4 sequential trials in pediatric ALL. Thank you, Persis.
Thanks very much, Savita, and thanks to the organisers for inviting me. I'm going to give you a brief overview of where we're at with CAR T-cell therapy for pediatric ALL and some of the challenges as we move forward. So, next slide, sorry. So this slide shows the data from major studies of pediatric ALL in turquoise and adult ALL in blue. And what's remarkable is that despite the use of different vectors, retroviral, lentiviral, different binders, different co-stimulatory domains, in over 300 patients with relapsed refractory ALL, the CR rate was 91%. Of which the overwhelming majority were MRD negative. Next slide, please. So what do we know so far? Well, we see unprecedented responses in chemorefractory ALL or patients who relapse post-transplant for the first time independent of tumour genetics, as long as we use this triad of lymphodepletion, second generation or above CAR T cells and short-term culture. The toxicity can be severe in patients with a high tumor burden. You can get severe CRS and neurotoxicity, but in children, this is generally manageable and reversible. Patients do relapse, and we'll talk a little bit more about that later on. And one of the important things to think about is persistence. And so there are some CARs that persist, in which case they're just a bridge to transplant, and so others that don't persist, and others that do persist, and hence can be a standalone therapy. And really, for this therapy to have an advantage over antibody-based approaches like blinatumomab, it's going to have to be a standalone therapy. Next slide. These are the survival curves from the pivotal Eliana multi-center study, which demonstrated the feasibility of delivering this therapy across multiple centers in multiple different countries. And based on these data, the EMA and FDA approved Kymriah, just letrozole, for relapsed refractory pediatric ALL. In the context of bone marrow relapse post-transplant, second relapse, and refractory relapse. Next slide, please. And these results from Eliana have now been corroborated with real-world data with Kymriah. This is the CIBMTR data published in Blood Advances last year, and it shows a 52% event-free survival in a cohort of 249 patients with ALL treated with Kymriah across a variety of centres within the US. Next slide. And if we compare these real-world data with Eliana, we see that the event-free survival at 1 year is very comparable. And if anything, there's rather lower toxicity in terms of severe CRS, which presumably reflects the fact that we're treating patients with a lower tumour burden and intervening early with tocilizumab. Next slide, please. So Saragarajan in the UK has shown very similar outcomes with our first 50-odd patients treated with Kymriah on a commercial basis. So with a median follow-up of just under a year, the 1 year event-free survival as defined in the Eliana study was 68%, which is, if anything, higher than the pivotal study. And even if you analyse by intention to treat, so the patients that take into account the patients that don't make it to infusion or don't respond, then the EFS is still good, 61%. What Sarah did was she then analysed the data with a composite EFS, taking into account molecular relapse and the need for further therapy if the CAR T cells don't persist. And then the event-free survival was around 34%. And actually, I think this is a much more genuine representation of where we're at with outcomes with CAR T cell therapy for ALL. So it's really about a third of patients that in fact benefit long-term from this as a single agent. Next slide, please. So what do we need in place to implement a CAR T cell program? So in the UK, we have a national MDT discussion of patient eligibility and alternatives. Obviously, you need an apheresis team skilled in lymphocyte rather than stem cell harvest. You need a cell therapy lab that's trained in cryopreservation and transport of the starting material and receipt of the product. You need pharmacy oversight. You need training of your nursing and medical staff, both in administration and in the complications of CAR T cell therapy, like CRS and ICANS. You certainly need access to an intensive care unit and specialist neurology. And within Europe, and the States, we have JC accreditation for centres delivering immune effector cells, and that requires a fair amount of documentation in terms of SOPs. Sorry, next slide, please. So Novartis have developed an onboarding program for new sites, and this has three major components around the apheresis and cell processing, the ordering process, and the traceability. They've developed a platform called Cell Chain Link, and the training of the individuals within the centre, and this typically takes around 9 months. Next slide, please. If we move to academic studies, of course, there are a number of additional requirements, and Dr. Chopwani has gone through this in great detail for what's needed in the States. Sorry, in India. You clearly need a GMP grade vector. That's incredibly costly and often rate limiting. There are waiting lists for development of vectors. You need an approved GMP facility and a validated manufacturing process. On the right-hand side of the slide, you can see the documents we generally require within the UK for regulatory and ethics submission, and that is quite arduous, particularly the IMPD, the dossier, and you need an experienced trial management team to deliver studies with what's called these 8 advanced therapy IMPs. And actually, this is very costly. And within the UK, it typically costs around £2 million to deliver a phase 1 study of 12 to 15 patients. So, I mean, the first time you do this, it's very difficult. We had a very similar experience to what Dr. Matthews was just referring to. Our first study, which we tried to do as a pan-European study, took 6 years to get from protocol to study initiation. Once the infrastructure and the templates are established, that process can be streamlined down to 12 to 18 months. Next slide, please. So I just wanted to spend a couple of slides talking about some important practical issues when you're treating patients with CAR T cells. You obviously need to ensure that the patient meets the eligibility criteria. There's no significant CD19 negative component. It's critical to give as little therapy as possible prior to the leukapheresis to preserve T cell fitness. This is a really important message, because if you give too much therapy, your T cells won't be fit, and then they won't persist in vivo. Ideally, you want a lymphocyte count of over 0.5, minimum 0.3. You want T cells of around 150 per microliter. And there are guidelines for when you should stop individual treatments prior to leukapheresis. So, for instance, we generally stop vincristine a couple of weeks before, and steroids at least 3 days before. But the earlier you can stop them, the better. Next slide, please. Once your patient's leukapheresed, you need to hold the disease for around 4 weeks until the product is made and shipped back to you. And one of the key things that we've learned is that the role of holding therapy is very different from the pre-transplant setting, where you want to try and achieve MRD negativity. Here, the key is minimal toxicity, particularly in the post-transplant setting. You don't necessarily need to achieve morphological remission, because CAR T cells will work in frank relapse. And you certainly don't want your CNS disease to break through, so you have to give IT therapy, but you need to give the minimum possible in order to reduce the potential for neurological toxicity, particularly in heavily pre-treated patients. So in general, both within the UK and within the US, we typically start with regimen A or B induction as holding chemotherapy. We give a couple of weeks and assess the response. And if that's not adequate, then we give escalating Capizzi without asparaginase. Next slide, please. So I'm going to switch to some of our research at UCL. So in collaboration with Martin Pule, we developed a novel low affinity CD19 CAR based on a different binder from Kymriah. It's called CAT19. And we showed in our preclinical studies that this resulted in increased proliferation in vitro and better tumor clearance and in vivo expansion in a xenogenetic mouse model. So based on these studies, in 2016, we started a clinical study, a phase 1 clinical study in relapsed refractory ALL in patients under 24. The primary endpoints of this study were toxicity and the proportion of patients who achieved molecular CR at 1 month. And it's important to note that this was a standalone therapy. We didn't consolidate patients with transplant. So patients received 10 to the 6 per kilo autologous T cells after standard flu-cy lymphodepletion. We treated 14 patients, a very heavily pre-treated cohort, median 4 lines of prior therapy, and 10 had relapsed post transplant. Next slide, please. So this slide summarizes the outcomes with the first cohort, which we published in Nature Medicine in 2019. We had a very good safety profile, no grade 3 to 5 CRS, nobody required tocilizumab. The main toxicity we saw was prolonged neutropenia in a proportion of patients. We saw excellent expansion and persistence of CAR T cells, and this mirrored what we'd seen in our preclinical studies. So the cumulative exposure to the CAR T was 5 times more than with Kymriah. The half-life of the CAR T was about twice as long. 12 out of 14 of these heavily pretreated patients achieved molecular CR. And at the time we published, the event-free survival was 46%, which is very similar to Eliana. Claire Roddy has done a study in adults which has shown a similar safety profile, and based on this, Autolus are now have licensed the CAT-CAR for use in adult ALL, and a registration study is ongoing. In both studies, the CD19 negative relapse was the major cause of treatment failure. Next slide, please. So given the complexity and cost of making a patient-specific product, it would be a major step forward if we could make an off-the-shelf product for multiple recipients. Next slide, please. And this approach has been pioneered by my colleagues Paul Vase and Wasim Kasim, who have used talons to downregulate the endogenous T cell receptor to prevent graft versus host disease. These are third-party T cells, and also to downregulate CD52, so you can use alemtuzumab to prevent rejection of the third-party CAR T cells. And proof of principle of this approach was provided by 2 infants who were successfully treated at our institution. Next slide. And based on this, Servier set up 2 studies of this approach in adults and pediatrics where there was no access to autologous CAR T cells. And so Ruben Benjamin has published pooled data from these studies last year in The Lancet. Toxicity was manageable, though viral complications were frequent because you're using alemtuzumab. What's clear is that you definitely need alemtuzumab. There was no CAR expansion or response in patients who weren't treated with alemtuzumab. But even in patients who were, the presence of CAR T cells was transient in most patients. And so the majority of patients were consolidated with transplant. And you can see the progression-free survival at 6 months was 27%. Given that these cells don't persist, the question arises, if they're not a standalone therapy, is this really any better than blinatumomab or inotuzumab as a bridge to transplant? And I think that question remains unanswered. Next slide, please. So I'm going to come back now to autologous CAR T cells. It's important to realize that Kymriah is just the first iteration of this, and Yescarta are the first iterations of this therapy. A number of challenges still remain. Around 50% of patients will relapse, mostly within 1 year. And there are broadly 2 classes of relapse. It can be due to limited persistence of the CAR T cells, in which case the relapse is CD19 positive, and that tends to happen early, or the leukemia can evolve to become CD19 negative, and that often happens a bit later. And the pattern of relapse depends on the nature of the CAR T cells. So you only get emergence of CD19 negative clones if you have persistent selective pressure, so you need the CAR T cells to persist. And that indeed was what was seen in the Eliana study, though actually in the real world experience, CD19 positive relapse has predominated. Next slide, please. One of the key causes of relapse is poor persistence of CAR T cells. We don't really know how long you need CAR T cells to persist in ALL, but an early study from Seattle showed almost universal relapse if you lose B-cell aplasia before 2 months and don't consolidate with transplant. So a number of factors feed into CAR T cell persistence. One is the design of the CAR T cells. Kymriah persists more than Yescarta, and that's probably because Kymriah uses a 4-1BB co-stimulatory domain as opposed to CD28. But 4-1BB on its own isn't sufficient for persistence. The choice of vector may be important. Lentivirus, most of the successful studies with long-term persistence have used a lentiviral vector rather than a gamma retroviral vector. And clearly the production methodology affects the CAR T cell phenotype. So in my lab, Luca Biasco compared the integration sites in circulating CAR T cells from a couple of patients with long-term persistence out to 3 years, and compared these to the selected memory subsets from the infused product. And he showed that 40 to 60% of the long-term persisting shared clones are derived from the stem cell memory compartment. And this suggests that this compartment plays a critical role in CAR T persistence, and that we should therefore design our methodology to favor less differentiated T cells with a shorter duration of manufacture, avoiding cytokines if we possibly can, and you can use small molecule inhibitors like AKT inhibitors. Next slide, please. Some patients lose their CAR T cells gradually over time, and this is probably due to senescence, but others lose them very abruptly, and this is likely due to immune-mediated rejection. So Cameron Turtle in Seattle showed no expansion and persistence after reinfusion of a second dose of CAR T cells in 5 patients with early loss, and this correlated with a cytotoxic T cell response against the CAR. Next slide. Shannon Maud from UPenn has investigated the use of a humanized version of Kymriah to prevent immune rejection of CAR T cells, and these data were recently published in JCO. Overall, 74 patients were treated, 41 were CAR naive, 33 had prior CAR T cells. The response rates were higher in those who were CAR naive, and the relapse-free survival at 2 years was 74% in the CAR naive patients, 58% in the retreatment cohort, but you have to take into account that only that's 58% of 64%. While the recovery of the proportion of patients having early B-cell recovery was lower in this study than reported with Kymriah of CTL109, there are still 15% of patients who have early B-cell recovery, so early loss of CAR T cells. So humanizing doesn't completely solve the issue of non-persistence, and intrinsic factors, as we were discussing, may be equally important. Next slide, please. Apart from early loss of CAR T cells, the other major reason for treatment failure is emergence of CD19 negative clones under the selective pressure of the CAR T cells. And this slide illustrates, this is illustrated on the fact box in this slide. You can see that at presentation, the leukemia expresses CD19. This is lost at relapse. And this can occur through a variety of different mechanisms, the most important of which seems to be CD19 negative mutations leading to a truncated protein which is not expressed on the cell surface any longer, so the CAR T cells can't see them. Next slide. It's important to note that the majority of CD19 negative relapses still express other B-cell markers like CD22, and this gives us an opportunity to target these cells. Next slide. So to address this problem of CD19 negative relapse, Terry Fry and colleagues at the NIH developed a CD22 CAR based on the M971 binder, and this indeed showed activity in a clinical trial. 11 out of 15 patients achieved CR. However, they observed a very high rate of relapse in patients who weren't consolidated with transplant, and they found that this was associated with downregulation in CD22 antigen density, not loss of CAR T cells, but downregulation. CD22, this study shows the feasibility of targeting CD22, but CD22 on its own is insufficient. And this led my group and others, by analogy with what's been seen with drug-resistant TB, HIV, to hypothesize that a single leukemic stem cell is unlikely to downregulate both CD19 and CD22 simultaneously. So maybe targeting multiple antigens might prevent relapse due to antigen loss. Next slide, please. So there's so how can we target two antigens separately? Conceptually, there are 4 different options, all of which have their own pros and cons, as outlined in the table. The first approach is to simply infuse different populations of CD19 and CD22 CAR T cells. The problem with this is that it's costly and complex to make 2 vectors and 2 atoms. The second approach in B is to use a single vector to encode 2 CARs, a bicistronic vector. So your single CAR T cell expresses both CARs. There's the potential for interference between the CARs, and we don't know what the impact of dual signaling would be on exhaustion or toxicity. The third option is C, where you co-transduce your T cells with 2 different lentiviral vectors encoding the 19 and 22 CAR. And the issue here is that you've now got a heterogeneous product with some single positive and some double positive cells, and that may vary between patients. And finally, if you're very clever, you can design your CAR so a single CAR recognizes both targets, but it is difficult to optimize that. And so all of these approaches are now being tested clinically, and I'll show you the results of the first generation studies of these approaches, including our own work. Next slide, please. By far the biggest study was published from the group in Wuhan, China, in blood. And in this study, they infused 2 different populations of CAR T cells recognizing CD19 and 22 in patients with ALL or NHL. The toxicity profile was pretty similar to Kymriah. The majority of ALL patients achieved a flow MRD negative CR, but the progression-free survival is 53%, which is the same as Kymriah. And of the 24 patients that relapsed, almost all of them, 23, relapsed with antigen-positive disease. And if you look at the persistence of the CAR T cells in the middle panel, you can see that 16 out of 24 patients had undetectable CAR T cells at relapse, and 19 out of 24 had B-cell recovery. And this suggests that poor persistence of the CAR T cells underlies relapse in this study. Next slide, please. The Seattle group, led by Rebecca Gardner, have taken a different approach using co-transduction with 2 different lentiviral vectors, including encoding CD19 and 22 CARs. Again, this approach is well tolerated, reasonable response rates. The issue here is that despite balanced populations of CD19 and 22 single and double positive populations in the product, the in vivo engraftment in patients was almost all the CD19 CAR T cells. And so poor engraftment of the 22 and bispecific CAR T cells really precludes assessment of the effect on CD19 negative relapse. Next slide, please. In a very elegant approach, Terry Fry and Krista McCall developed a tandem CAR recognizing both CD19 and CD22, and the Stanford group have tested this in 2 parallel studies in adults and children. The adult studies recently been published in Nature Medicine. Again, they used lentiviral transduction. Interestingly, that here they used closed manufacture on the Prodigy system that Dr. Matthews was referring to. Standard lymphodepletion, a good safety profile, and reasonable CAR T cell expansion and persistence. Majority of patients, 14 out of 17 ALL patients, went into CR, but 10 of these relapsed, of whom 5 had CD19 negative or low disease. So the median progression-free survival was disappointing at 5.8 months. Next slide, please. Interestingly, the 5 patients who relapsed with CD19 negative disease all still had expression of CD22, which raises the question of why didn't the CD22 element of the TANCAR prevent relapse? So the authors went on to show that when stimulated with targets expressing CD22 alone, bottom left-hand panel, the tandem CAR T cells secreted less cytokines than T cells from the same individual transduced with a monospecific CAR recognizing just CD22. And so this illustrates that optimal engineering of a TANCAR is very difficult, particularly for 19 and 22, where 22 is way out from the cell surface, whereas 19 isn't. Next slide, please. The approach we took was to collaborate with Autolus, who developed a bicistronic retroviral vector encoding 2 second-generation CARs with novel binders. Both of these binders were humanized to reduce immunogenicity, and to facilitate broader application, we used a gamma retroviral vector and automated manufacture on the Prodigy. And those were commercial decisions, but actually they have had an impact. And so you have to take these sort of changes into account when you're designing your study. Next slide, please. We tested this approach in a multi-center phase 1 study called Amelia with dose escalation at 3 dose levels. Patients received standard fluci, lymphodepletion, and depending on the leukemia burden at the beginning of lymphodepletion, CAR T cells were infused as a single dose or as a split dose in patients with higher tumor burden. Next slide, please. So these results are in press and should come out fairly shortly in Nature Medicine. Again, we saw a very favorable toxicity profile with no severe CRS, just 1 case of grade 3 neurotoxicity. Overall, 13 out of 15 patients achieved an MLD negative CR, and the swimmer plot shows the outcomes in more detail. 9 patients relapsed, the majority with CD19 positive disease, and the 1-year and event-free survival was 32%. Next slide. So why didn't dual targeting prevent relapse? And this slide shows the persistence of CAR T cells as assessed by qPCR in the blood. And you can see that while the initial expansion at 1 month of the CAR T cells is pretty similar to what's reported for Kymriah, by day 100, the majority of patients have lost their CAR T cells. And so this suggests that poor persistence underlies relapse, and this correlates with a more differentiated effector memory phenotype in the product, which you can see on the top right, where there's a lot of those yellow cells, which are effector memory cells, and that may reflect in turn our production methodology, which required cytokines and used the beads that go along with the Prodigy system. Next slide, please. So in summary, this study demonstrated the feasibility of simultaneously targeting 19 and 22 with the bicistronic CAR, and that this approach was well tolerated with no excess toxicity compared with single antigens. We saw a high initial remission rate, but the relapse rates were really no better than Kymriah, and the commonest cause of relapse was due to loss of CAR T cells, which we think is associated with a differentiated phenotype of the product. And again, this precludes assessment of the impact of dual targeting on antigen-negative relapse. Next slide, please. So are we there with bispecific T cells? No, probably not, but we're a bit closer than we were, and I'm pretty convinced that ultimately this approach will overcome the problem of antigen-negative relapse. We've shown that dual targeting is feasible without excess toxicity. The optimal strategy has yet to be defined. So far, none of the strategies tested has shown a clear advantage over Kymriah, but we have learned 2 important lessons. First, persistence of CAR T cells is a prerequisite for assessing the impact on antigen-negative relapse. And second, for a CD22 CAR to be effective, it requires activity at low antigen density, and this may be challenging, particularly in the TANCAR format. So where to next with the bispecific CAR T cells? Based on the lessons that we've learned, We're currently combining two elements that we believe are critical. I showed you the excellent persistence of our low affinity CD19 CAR, and we're now combining this with a novel CD22 CAR that recognizes low antigen density, down to 250 antigens per cell. And we're using the same methodology we used in cohort 1 of CARPAL, which was associated with good persistence. So we think that the presence of Cat19 cells should ensure persistence, and it should be at least as good as Cat19 alone, and hopefully there's 22, if they persist, will prevent antigen negative relapse and vice versa. Now, I can't show you the data from that study. As yet, we've treated 6 patients, but I think the initial data are extremely encouraging, and I think there's a real chance that this will represent a significant advance over Kymriah, and will demonstrate, we'll be able to work out whether dual targeting prevents antigen negative relapse. Next slide, please. So I hope I've conveyed to you that, so next slide, please. So I hope that I've conveyed to you that while Kymriah may be licensed, there remain unanswered questions. We're just at the start of this new treatment modality. The big questions remain about the impact of vector choice, lentiviral versus gamma retroviral, automated production. Can we do that successfully without compromising the persistence of the CAR T cells? How can we manipulate our production methodology to preserve this key stem cell memory population? We've talked about how we can prevent relapse by improving persistence and targeting multiple antigens. And then one area I haven't touched upon is the placement of CAR T cells. Can we avoid transplant in high-risk relapse? Can we use them in patients who are resistant to frontline therapy? And this is being assessed by the Cassiopeia study. And how do we optimally combine CAR T cells with other treatment modalities like blinatumomab, inotuzumab? And to answer these questions, we're going to continue to need randomized clinical studies, both in the commercial sphere, but also in the academic sphere. So I'll stop there, and thank you all for your attention. I'm happy to take any questions.
Thank you, Dr. Amrolia, for this excellent talk. Now we'll open the floor for questions. The first question is for Dr. Geeta. So Dr. Geeta, the question is, what is the harmonization of the Indian stem cell or the gene therapy guidelines with that of FDA and or in EU or the European Union?
So the harmonization is basically on the definitions as well as the process of regulatory approvals and also the ethical considerations. So it's kind of a global, as I said, that there are many global challenges related to stem cell research and therapy, and they are almost similar in most of the regions.
Thank you, Dr. Geeta. Next question is to Dr. Matthews. Dr. Savita, you were saying something?
Yeah, I was just going to say, sorry, I didn't realize I had my mic muted. So Vikram, how is the dosage of CAR T cells calculated during any cancer treatment?
As far as CAR T cells goes, there's been a lot of early phase trials, and anything between 2 million seems to work, and people have kind of worked around those doses. We obviously don't want to go too high because we already worried about cytokine release syndromes and toxicity. And I think Dr. First's talk was excellent, where he showed the concern when there's significant disease burden, you may actually want to split up the dose into 2 doses rather than give the whole single dose. That's my understanding. Maybe I think Dr. Amrolia will probably have some better insights on upper limits.
It's an interesting area. It's very different for a conventional drug. So there's no clear association between, once you reach a threshold, which is probably around 10 to the per kilo, there's no clear association between higher doses and efficacy, though higher doses can be associated with increased toxicity. You've got to remember that these CAR T cells, you're putting in a tiny dose of T cells, talking generally about a few mils. And when they go into the patient and they see antigen, they expand thousands or tens of thousands of fold. So whether you infuse 1 million or 3 million is to an extent largely irrelevant. You probably shouldn't infuse too many, otherwise you will get severe CRS.
On the same lines, Persis, there's a question here which says that CAR T cell associated toxicities are manageable and even reversible in case of children. What makes it easier to control these toxicities in children rather than adults? And is that really true?
It's a good point. Well, I don't think we really know that, but it is true to say, it's also true in the transplant sphere, isn't it? That the children are generally more robust than adults, so they can tolerate the hypotension associated with CRS. They can recover from neurotoxicity better than adults. But it is a good question. I don't have clear answers. I think there are ways around this by fine-tuning your CAR T cells. Then I think that it is likely that these technologies will be appropriate for adult patients with ALL too.
Okay, thank you. And the next question is to Dr. Jotwani. Given the very high cost of gene therapy, it will be good for Indian scientists to collaborate with scientists abroad for technology development in India. Has ICMR considered a suitable regulatory framework to facilitate such collaborations leading to clinical trials and also to share the intellectual property rights?
Yeah, ICMR welcomes the international collaborations, but it depends on the phase of the trial or the phase of the gene therapy product development. So any such product coming to India will require, it is prerequisite to have a clinical trial. If the product is brought to India, they have to have a clinical trial or the approval from the country of origin. Okay, but if you are developing a product in India, then you have to follow the Indian regulations.
Okay. Thank you, Dr.
I'm
Geeta.
not sure about the cost effectiveness of these products if they are developed and sold outside India. We are more concerned about Indian patients, how it is cost effective for Indian patients. That depends on the IP, definitely.
Okay, thank you.
Yeah.
Vikram, what are the recurring challenges you faced in clinical setting, in the clinical setting in treating patients with CAR T therapies?
So I've really not treated patients at this point in time. We've done our preclinical validation runs. We're still awaiting the CDSCO approval to start our CAR T trial. Obviously, unless we get that, we can't treat because we can't possibly afford Kymriah or etc. As a commercial product, and neither are they interested in doing clinical trials in India at this point in time. So we just have to wait for approvals to do our study.
And by the way, would you know what the cost of production of CAR T would be in Miltenyi?
Well, it's not Miltenyi, it's within our costing structure. Their consumables, etc., will be a certain fixed cost. I think, and even that there is a certain differential pricing. What we have worked out is it works out to around 25 to 30 lakhs will be the cost to manufacture. But I think there is significant scope to actually research in this area. There's a lot of areas like, the use of the consumables. There's a lot of redundancy in the system, which actually you can work with to further reduce the cost. That's my sense at this point in time.
Okay.
So for example, the amount of antibodies, the media, you really don't need to go to 14 days expansion. You can cut it down to, you get enough cells, at a very short period of time. And as Dr. Amroli also kind of referred to, you may actually want to do it earlier because the kind of the cell subset profile may actually be more favorable early than late.
Okay, Thank you, Dr. Vikram. The next question is to Dr. Amrolia. Migration of CAR T cells into tumor sites may not be sufficient enough to ensure tumor destruction because of the immunosuppressive nature of the tumor microenvironment. Hence, what are the approaches that can be taken for improving CAR T cell infiltration into the tumor microenvironment?
Yeah, it's a very good question. We're very lucky with ALL in that it's a liquid tumor, and I think that was quite fortuitous for this version of CAR T, for CD19 CAR T cell therapy. It's a much higher bar to extend these therapies to solid tumors, because you do have to overcome the problem of the tumor microenvironment. And obviously, Yescarta is licensed for diffuse large B-cell lymphoma and others. But as we move to other malignancies, I think that is going to be a particularly difficult area. And there are a number of things that one can do. You can, this is you're engineering CAR T cells, and you can. It sounds fanciful, but you can do a lot of additional manipulations to enhance the homing of your CAR T cells to the tumor. If you know the microenvironment issues that are switching off your CAR T cells, like secretion of IL-10 or TGF-beta, you can engineer ways of manipulating your CAR T cells to put in dominant negative mutations that can overcome those limitations. You can treat with, if the problem is exhaustion of CAR T cells, you can treat with checkpoint blockade inhibitors in addition to CAR T cells. People have spent a lot of time thinking about these and trying these different modalities. At the moment, the biggest block to extending CAR T cell therapy to solid organ cancers is the target. In hematological malignancies, we have very well-defined surface antigens that tend not to be expressed in the rest of the body. But identifying specific targets like that in solid organ malignancies has been really quite challenging, and then you have to overcome all these other issues that I've mentioned. So it is true to say that so far none of despite a lot of effort, actually, people haven't successfully extended CAR T cell therapy to solid organ cancers yet. But I think that will come. It will just take some time.
I have actually a couple of questions for Geeta Jotwani. Could you please advise on what special informed consent form may be required for CAR T cells or for gene therapy? And secondly, what is the approval time for gene therapy clinical trials? Are these steps, can they be done in parallel?
Yeah, so regarding this second question, I'll answer first. Regarding gene therapy clinical trials approval, yeah, it has to be done parallelly. You have to submit a clinical trial application to CDSCO, and GTAC has a recommendatory role that it will technically evaluate and give their recommendations to CDSCO, and CDSCO with the consultation with their subject expert committee finally evaluates the clinical trial protocol, and they'll consider the recommendations of ICMR committee, that is Gene Therapy Advisory and Evaluation Committee. That is how the framework has been proposed.
With regards to ICF?
ICF, yeah. I said it is a thought process, and we are looking forward to evolve a separate ICF for gene therapy products or the participants of the clinical trial, because it's a new modality and it does require a different kind of informed consent, because a lot of risks, I mean, looking at the ethical and legal aspects of it, it is important to have coined a separate and a different ICF. And we are working on that and will be soon there.
Thank you.
Thank you, Dr. Geeta. And the next question is to Dr. Vikram. So the question is, considering the limitations involved in the development of an ideal vector and also the production process in place, what is the scope of getting the cost of CAR T cell therapy to an affordable range, particularly in developing countries like India?
Yeah, so I think that's a very valid question. So if you can de-link the vector production and the CAR T cell manufacturing, which is what I think is one strategy to reduce the cost, will be important. We are focusing on the CAR T cell manufacturing and standardization, and we think that this is a model that can be replicated in multiple tertiary centers in the country with a certain infrastructure cost, but will be way less than trying to set up centralized manufacturing processes with the semi-open system. And even within these systems, for example, right now, Miltenyi Prodigy has kind of got a monopoly on it, but there are newer closed systems that are being developed that are even less expensive. So it's an area that we can research on and can also fund research to develop closed systems to do this kind of transduction. So it's an area for potentially active research. With regards to vector production, yes, it would be nice to have completely indigenous vectors, but I think as is being done by many industries, they are inline licensing these vectors and bringing it to the country. So it all depends on how that's going to be costed. And for that, I really believe the more the competition we have, the costing is going to significantly reduce on its own, just the vector by itself.
Sure. Thank you, Dr. Vikram. I think perhaps there is time for one last question, and that is to Dr. Persis. Should we do a pre-immunotyping phenotyping in autologous CAR T cell therapy and modify our approaches based on the T cell phenotype and the activation status, like introduction of a suicide gene or manufacturing process, etc.?
Yeah, it's an interesting question. I think the thing to do is actually to optimize your manufacturing methodology and assess the phenotype before you ever go into your clinical study. So you need to try and modify the way you make the T cells to preserve TSCM and TCM in your product. And as I mentioned, there are a number of ways that one can do that. In terms of suicide gene therapy, I think that has a limited role. It might make sense if you've got a patient with very severe CRS. Well, why don't we just have a suicide gene and be able to switch it off? And of course, there are things like ICAS-9 that one can use to do that. But the trouble is, once you've switched off if it's a suicide gene, once you've switched off your CAR T cells, the TAR T cells are gone, and then your patient's going to relapse. So, as I keep saying, I think persistence is key if this is going to be any better than blinatumomab. So suicide gene therapy probably isn't the way to go, but what people are developing is inducible CAR T cells, where you can use a drug to modulate the effect of the CAR T cells without deleting them. And so you make the CAR T cell quiescent when you give the drug, and the CAR T cell switches off again when you take the drug away. And that may indeed be a way of overcoming toxicity. For me, I think the toxicities within ALL, with CD19 CARs, are not the biggest issue. I think we can handle those. The biggest issues are cost, so that we can expand this to much larger numbers of patients, and how do we avoid relapse by making the CAR T cells persist and avoiding antigen-negative relapse.
Thank you, Dr. Ambroglia. I think I would like to thank all the speakers in this session, Dr. Jotwani, Dr. Matthews, and Dr. Ambroglia, for their excellent talks and insightful discussions. I know there are several questions that we have not been able to answer, and these will be shared by the ASGCT to the speakers and then emailed back to the registered participants. So now we move on to the next session. Initially, there was a break plan, but in the interest of time, we will continue to the next session. And that's going to be on emerging therapies in lower middle income countries. The first speaker of this session is Dr. Federico Mingozzi. Professor Federico Mingozzi is one of the pioneer gene therapists with more than 2 decades of experience in gene therapy, immunology, biochemistry, and molecular biology, both in an academic as well as in an industry setting. Currently, he is the Chief Scientific Officer at Spark Therapeutics, and he has developed several first-in-human clinical studies for gene therapy while serving as the Director of Translational Research at the Center for Cellular and Molecular Therapeutics at the Children's Hospital of Philadelphia. So today, he will give his perspectives on the current challenges in the development of gene therapies. Dr. Mingozzi.
Thank you, Jay, and I want to thank the organizing committee for inviting me to this symposium. I assume you can see my slides.
Yes, we can see. Thank you.
Great. So in this presentation, I thought I would give you an overview of in vivo gene therapy with AAV with some examples of what we've learned, current challenges, and also the experience of Spark Therapeutics and before the work we've done in academic centers that really was led to the development of a gene therapy all the way to the market approval. So I think based on the previous presentation, we know that when we are thinking about gene therapy in general, but also for in vivo gene therapy, the ideal goals are having a therapy that involves a single or very few vector administrations that are therapeutic in terms of expression of the gene of interest. And then, of course, we want always a long-term acceptable safety and efficacy profile. I've been working for basically my entire career on the development of gene therapies based on adeno-associated virus vectors or AAV vectors. These are viral vectors that are derived from a virus that is commonly found in nature and actually also infects humans. And this virus is engineered to carry a transgene expression cassette. So all the sequences necessary to express a gene of interest in a target cell. Recombinant AAV vectors that we use for gene therapy are quite good tools for the purpose because they are replication defective. They don't have any viral coding sequences, and they can infect post-mitotic cells, which may persist over time and lead to long-term efficacy or transgene expression. They're mainly non-integrating, and again, I will give you some examples on how there's a lot of data emerging that with this tool we can truly develop safety and effective therapies. This comes from preclinical studies and also human trials. AAV vectors have been used to target many organs, which I listed here. I took this picture from a review article, and depending essentially on the kind of AAV vector we use in terms of serotype, but also the route of delivery, we can target the eye, the brain, spinal cord, motor neurons, for example, and intravenously, this is the route that has been used to target both the CNS, the muscle, and the liver. I want to start from an example of liver gene therapy, just because this was something that we've worked on for a few decades. And in fact, when it comes to the liver, this is quite an attractive target to, for example, develop gene therapies for clotting deficiencies. And so I went back to the literature and I was able to find this paper published in 1997, so a little while ago, as you can imagine, where there was one of the first proof of concept in mice that we could target the liver with AAV vectors to express human factor IX. So this is the protein that is missing in hemophilia B. And this was a long-term expression. Now, then if you look at the literature, of course, there were many studies also conducted, the pioneering studies conducted both by us at CHOP, but also Dr. Natwani Taddenham at the UCL. And then in 2017, there was this 20 years later, 20 years of development, as you can imagine, there was this manuscript. This is work from Spark that we published showing long-term expression of human factor IX variant that is called Padua in humans, showing also that the expression of this factor IX from the liver following AAV gene transfer was leading to long-term correction of the disease, as is shown here in the panel on the right, where you can see that prior to gene therapy, the subjects enrolled in the trial experienced bleedings, even though they were actually treated with recombinant protein. And then after gene therapy and after interruption of the conventional protein replacement therapy, then the subject had virtually no bleeds. So this is important because this product, as long as also products developed by other biotechnology companies, are currently in phase 3. And so we would expect that these will become approved therapies for the treatment of hemophilia B. And at the same time, this is a slide that I made taking a table from a review article published in 2018. There's also other therapies ongoing, other gene therapies in development for hemophilia A. We also have one in development. And so also these therapies are some of them are in phase 3. What is important about this slide is that I like the fact that it gives you a nice timeline of the development of AAV gene therapy, starting, of course, from the essential identification and isolation of the genes that are at the base of genetic disorders, right? And so the main point here is that to get to where we are today, we had to address key challenges. One is the manufacturing challenges, which I will talk in the next slide. And then, of course, platform challenges, which again, I want to discuss in the next following slides. For manufacturing, the manufacturing of AAV vectors, and this in the slide is just an example. So there's different platforms that you can use to make AAV. I would really only summarize manufacturing for AAV by saying that these are very complex biological entities, right? Because we have a protein capsid that carries a nucleic acid genome. And so making AAV is not trivial and has been an important barrier to entry for the field of gene therapy. Moreover, and this is again a graph that I was able to find in the literature. As you can see, this is the y-axis here are basically genomic particles. So how many vectors we have to make to carry let's say, initial studies, toxicity studies, and then initial early trials all the way to supply, let's say, commercial supply to sustain a gene therapy drug, right? To commercialize a gene therapy drug. And so you can see how, when we are dealing with rare diseases, of course, the numbers are lower, but then as we hope in the future, we will go to more prevalent indications and we will see gene therapy becoming a more mainstream, let's say, therapeutic modality, then the manufacturing challenge is something to keep in consideration, along the fact that manufacturing of AAV vectors is quite expensive. So the cost of goods contributes to the high cost of these drugs. When it comes to the platform challenges, I took this table from a report from McKinsey and Company, which is, as you can imagine, not quite scientific, but I think it captures this essence of the current challenges of the AAV platform. In fact, what we are still, in a way, trying to figure out is the fact that, of course, we're using a modified virus to develop AAV vectors and to transfer genes to the target cells. And so then our immune system doesn't quite like them, right? And so getting past the obstacle of the immune system still remains one of the most important developments for the platform. Along that, other important challenges are the fact that we want to develop vectors that are quite targeted and allow us to achieve therapeutic efficacy at doses that are not so high, let's say. I mean, this is a kind of a generic statement, but I can actually link this statement to the previous slide, right? The lower is our therapeutic dose, the less will be our manufacturing challenge. And then, of course, in some cases, and particularly if we think about trying to make this therapy mainstream, we want to be able to control the expression of our gene of interest, both spatially and also temporally. And then, of course, there's a number of attributes that you can address or target to then achieve these goals. When it comes, and I promise I will not go into the detail, when it comes to AAV vectors, I think I try to summarize here the different components, starting from the very vector where we have an AAV capsid, the promoter that drives the expression of your gene of interest, the genome and delivery method. And then, of course, there's manufacturing. And so there's a number of desired property that we, meaning the field, are working towards to really evolve the protein and bring these therapies to the next level where we actually are going to be able to hopefully target multiple diseases successfully. These are the take-home messages. I think I don't need to go in the detail of that. I think I capture the fact that there's promise. The manufacturing is complex and expensive, and this is an important barrier to entry in the therapy. And there's some technology hurdles which we're trying to address. But then the question is, can we actually develop? I was trying to. Sorry for the audio, which started automatically. Can we actually develop drugs based on AAV vectors? So I wanted to give you the example of the development of a gene therapy for Leber's congenital amaurosis, which is a disease of the retina that is caused by a very defined mutation in the RPE65 gene. So this is an ultra-rare disease and is characterized by retinal degeneration and other symptoms related to poor vision, particularly in conditions of lower light. So one of the problems that patients also experience is the nystagmus, which is basically the inability for them to maintain the pupils fixed to a specific point. So this drug, we started the development back in 2007, the first phase 1/2 trial at Children's Hospital of Philadelphia. Then the phase 3 was taken by Spark, and today is a drug that is called Luxturna that is approved in US, in Europe, and many other countries. And these approval actually keep coming quite a long time, and I hope there will be a drug available worldwide. Now, the approach here is basically consisting in the injection of an AAV vector encoding the missing or the non-functional gene, the RPE65, subretinally. And basically, there's been a lot of studies, of course, conducted, but I think one of the important ones is that in the natural occurring animal model of RPE65 deficiency, the dog model, there was a very good efficacy profile. As you can see, 35 dogs were treated and 34 actually had efficacy. And the dog in the picture, actually, his name was Lancelot. He was actually received the treatment. The vision was restored for more than 10 years, actually, until the dog passed away of naturally. So this is an example of the clinical development that went into the RPE65 program that led to the approval of this therapy. There was a lot of work done in the subject population, which included the collection of a natural history, because that was missing. We're talking about a rare disease, ultra-rare disease, and we're talking about blindness. And at the time, before gene therapy came, and advanced therapy came for blindness, essentially blindness was simply a disease with no treatment. So there was not a natural history, not a lot of natural history of the disease itself. So there was a big effort about collecting natural history. And when it comes to the clinical trial, there was an initial trial in a small number of subjects, 12 subjects, which was a dose escalation phase 1/2 trial, first treating one eye and then treating the contralateral eye, which then led to the identification of the key endpoints in discussion with the regulatory agencies that would be used for regulatory approval. And one of them was this multi-luminance mobility test, or MLMT, which was a new test which had to develop and validate it. And the validation of this test was actually done in a subject with inherited retinal disorders, a normal subject, to really understand if this test could divide the two populations, visually impaired and normally sighted people. So all of that led to a phase 3 study, which was then conducted in 31 subjects, and then finally the approval. So these are the milestones of this therapeutic, of this project, starting from, of course, again, the identification of the gene, the early proof of concept that I talk about. And then 2007, I say this phase 1 was initiated at Children's Hospital, Philadelphia, and then continuing up to the phase 3, which was then conducted by Spark, and then finally the approval in 2017 for the FTAs, later on for EU and other countries. The phase 3 trial, real quick, was a randomized trial where subjects were enrolled in the study and then randomized to a treatment group, intervention group, and a control group. And then the design was a crossover, meaning that after 1 year of follow-up, the control group would be actually rolled into the intervention group, which will receive the actual therapy. So one little words about the main endpoint that was used for approval, which was very complex because we had to define it and actually validate that. The multi-luminance mobility test was a test that was essentially an obstacle course, and I'm gonna, an obstacle course conducted at different levels of luminosity, starting from a very, very little light level, those characteristic of a summer night without moon or indoor night light. And just as a reminder, the subject affected by this disease have a very hard time to navigate an environment in low light level, all the way to very bright light. And so each one round of testing for this multi-luminance mobility test would imply subjects conducting the or trying to going through the MLMT, so the obstacle course, and then there was a scoring based on time to get to the end of the obstacle course, and also errors made in the way. So this is an example. I hope this will start. Okay, perfect. And so you can see how this is the same subject, basically a baseline and one lux light level, which and you can see how before therapy the subject had a very hard time to do the to perform the MLMT test. In fact, could not do it, and you can see on the right of the slide how after the therapy there was a significant improvement in the ability of the subject to get to the end of the obstacle course. And so essentially the key here was to take these videos and transform them in something that looked like this. So this is a graph that shows you the score change after therapy. And so the green light would start, and it's not represented here, will start essentially at the same level of the blue line, which is basically the two groups in the phase 3 trials, so the intervention group and the follow-up group. And you can see how after gene therapy, these subjects had a dramatic improvement in their scoring on the MLMT, and then the delayed intervention group, so the control group, upon rollover into the intervention group, also had an improvement in the MLMT. And recently we presented that up to 5 years after gene transfer, we still see the improvement being stable. Now, these are the other endpoints. I don't need to go into the details, but a lot of the additional endpoints from the phase 3 trial turn out to be significantly affected by the therapy, such as light sensitivity, some other endpoints, and visual field. However, for instance, visual acuity in this case was not significantly changed after gene therapy. So these are some of the side effects related to the therapy, which are mostly related to the fact that the subretinal delivery of AV vectors is quite an invasive surgery. So it does have some complications. Now, innovation in gene therapy, of course, there's a lot of technology innovation, manufacturing, as I told you. However, what we learn is also that that doesn't stop here, right? Because to commercialize a drug it requires a lot of innovation. Now, this slide there's a lot of points that I want you to see, and then I will discuss just a few. Of course, we are talking about, as I told you earlier, ultra-rare disease which had no cure previously. So patient identification is a major task for RPE65 deficiency and in general inherited retinal disorders, because there's no treatments out there, although the landscape of that is changing. So we had to establish a system to essentially identify patients and genotype them. And then, of course, there's all the part of market access that is complex and it's quite complex. And one of the things I want to say is that even for when it comes to pricing reimbursement of the drug, we had to think about innovative ways to essentially get to secure coverage. So in the US, let's say the choice was that there was the possibility essentially to have a rebate if, for example, the therapy would not provide efficacy after treatment. So a partial payment back. I also wanted to add some comments about some country-specific considerations, and to prepare this slide, I reached out to my colleagues in regulatory and clinical operations, and I would say this is something general considerations more for thinking and discussion, which are that in lower middle-income countries, they tend to be considered more for a phase 3 trial versus phase 1/2. I think it's because phase 1/2 trials tend to be very small, as I show you in my previous slides, and so there's a need for a tight control on all the variables that can get into the trial, and high consistency. Otherwise, meshing up safety and efficacy in a small trial, then the risk is that you don't get any conclusive results. So then that's the first consideration. Of course, other consideration involve the framework and timing for regulatory approval as well, legal and ethical considerations, which include liability and intellectual property. I'm closing here. I hope my presentation was helpful by saying that the AV platform can support the development of gene therapies for a variety of diseases, starting from, of course, genetic diseases, and then acquired will come, hopefully. There's a lot of innovation that needs to go in manufacturing platform, which really hopefully make these products cheaper from a cost of good perspective and more reliable. And then I think there's also a lot of learnings from the clinical development commercialization that again will help future developments. So I want to stop here and thank you for your attention.
Thank you, Dr. Mingosi, for this inspiring talk. We will take questions at the end of this session in a panel discussion. So our next speaker is Dr. Savitha Rangarajan. She is an acclaimed clinician in the field of clinical care and research in hemostasis and thrombosis. She is currently an associate professor of clinical hematology at the University Hospital in Southampton and also the director of Southern Hemophilia Network. Her main interest is in the management of complex disorders, coagulation disorders, and in particular patients with inhibitors and the delivery of cost-effective models of care for all patients with bleeding and thrombotic disorders. Dr. Rangarajan is keen to mirror the model of hemophilia care within the NHS in other countries, particularly in LMICs such as India. Today, she will discuss on the emerging therapies in India. Dr. Savitha Rangarajan.
On how, sorry, I didn't unmute myself. Thank you, Dr. Rao. And on my talk is not very scientific, but it's just my thought process on where we are and where we will be in India with gene therapies. And for me, emerging therapies is about more about gene therapies. So gene therapies includes in vivo, ex vivo, and gene editing technologies. The target diseases at present are focused mainly on rare inherited diseases, and I will explain about the rare diseases in my later slide. However, creativity in gene therapy may help many other chronic conditions, as Federico just mentioned before, and perhaps in oncology, as we saw Jayendran's presentation as well. We believe that gene therapies will be mainstream, and I am using the same words as Federico used, in a few years as therapeutics move up from protein to DNA. As I mentioned before, the continuum of gene therapies include in vivo gene therapy, where we just infuse the virus into the patient either by intravenously or into the subretinal space. And of course, we have gene editing, which we haven't actually had any at the moment, at least not in hemophilia, and cell therapies where one would take the cells out and then introduce the gene and transplant the gene, particularly for rare diseases, which of course happens for sickle cell and thalassemia, for example. The definition of rare diseases is different in different countries, as you can see in this slide. For example, in Europe, it is less than 5 per 10,000 population, and in Australia, less than 1 per 10,000 population. And I've taken this from the National Policy for Treatment of Rare Diseases in India. If we apply the international estimate of 6 to 8% of the population being affected by rare diseases, we would have a tremendous amount of, we would have a great number of people, 72 to 96 million people affected by rare diseases, which is a significant number. However, it's just it's a guesstimate because we really do not have prevalence data at present in India. Only about 450 rare diseases have been recorded in India from tertiary care, although worldwide there are significantly a large number. And the most common rare diseases include hemophilia, thalassemia, sickle cell anemia, and of course, other conditions like Pompe's, Gaucher's, cystic fibrosis, and certainly the muscular dystrophies and spinal muscular atrophy. So my talk, as I said, it's not very scientific, it's just my thought process. And where are we in gene therapy and can we fast track? I'm going to talk a little bit about science behind gene therapy, research and preclinical evaluation in India, our biotechnology industry and potential to scale up easily, regulatory agencies. Of course, Dr. Geeta Jotwani has already spoken quite a lot about it. I'm also going to talk about site readiness, where we can do clinical trials. And for me, most importantly, is patient engagement. And I think I'm going to actually do my talk reverse, where I'm going to start off with patient engagement. Now, this was in popular press, talked about spinal muscular atrophy and the drug Zolgensma, where it was quite a big news that a drug that can save lives of kids cost rupees 16 crores, which transforms to $2.25 million. And what can India do? Then we did have a good story to tell, where a child was actually treated with Zolgensma and it was bought at 16 crores because they managed to raise the funding through crowdfunding to buy the medicine. And I think this has been going around through a lot of WhatsApp messaging and of course in popular media as well. So therefore we come to the point where gene therapy hope or hype in India, and I certainly hope I certainly feel it's going to be hope rather than hype. And then when I looked up the internet, I did see this particular quote where the government sets up rare disease crowdfunding portal. And I think that's something that is good to see currently, but I'm sure we should be able to get gene therapy at a cost-effective price and model. Now as I said, I'm going to talk about patient engagement. And for us as doctors, obviously, and for clinical trials, safety is the most important thing. And for us, do no harm is critical. I say this particularly in India because, clinical trials have had bad press in India, and where unfortunate incidents may have taken this, where patients were treated as guinea pigs, that's the optics. And I hope that doesn't come. And I'm sure there's a lot of governance structures that have been put in place, as Dr. Geeta Jotwani has already alluded to, and I'm sure will not be there again. I'm going to play a video of some of my patients' voiceover of the clinical trials for gene therapy that they took part in.
My main reasons was for my family. I've got a daughter with the carrier gene, and I was concerned for her future and my future grandchildren. So I wanted to try and see what I can do and make a difference to help improve their lives.
I felt as if it was my duty to do it. I think it's something that needed to be done. As you know, when I grew up, I spent the majority of my childhood in hospital and in bed recovering from a silly little bleed that I don't remember doing. And while in there, I'm always thinking to myself, oh, I wish I was like the other kids. Growing up in South Africa, the sports there are quite big: rugby, cricket, football, and why can't I do that? Why am I here? And I don't want the future children to ever go through that, ever. And if taking part in this phase 1 study is the first step for that future, I'm all in.
I was brought up to know that this was an incurable condition, and now, in theory, I'm cured. It's amazing to actually be part of that and to actually be one of the first to experience that. I had hemophilia all my life, and I grew up with it just being there. I didn't know any different, and now I didn't realise actually the effect it had on my life before. Simple things like taking the medication, being very conscious about what you're doing.
As a haemophiliac, every day is a risk. Every day you're taking a chance when you're walking across the streets or you're just going shopping. Anything could happen to you. So when you're given the opportunity to take a risk and change the world, why not? Why not be the first? Why not be the 10th? My mother always said to me is be the change you want to see in the world, and I believe it starts with you.
So being a hemophilia treater, I clearly would talk about hemophilia. And of course, as you all know, it is a royal disease. Starting off, we know that Queen Victoria was a carrier. And of course, the Tsarevich Alexis was a hemophilia patient. Being a royal disease, it also comes with its own cost. And of course, hemophilia is a very expensive disease. And I would say that 70% of the hemophilia population across the globe actually do not have access to good treatment, even at the present time. For me, what is very important is that there was a recent publication actually from the UK where improving patient-informed consent for hemophilia gene therapy, and they called for a case for change. I think it's important to understand that we need to take the consent process for gene therapy very, very seriously. And the regulators responsible for approving medical products have published guidance on informed consent for physicians and pharmaceutical manufacturers in hemophilia, including for gene therapy. Recommendations have been made about best ways for patients with hemophilia to discuss gene therapy with their physicians. Yet, poor communication of complex topics, such as gene therapy, can be problematic, especially if the patients lack the skills and confidence to understand and discuss science, or for physicians with limited time in clinic. Now, I think this is extremely relevant for our country. As you know, hemophilia treatment advances have always been driven by safety because of hepatitis B, hepatitis C, HIV, etc. We have improved our therapies for hemophilia. And of course, it was always called the beginning of a cure when we had a lot of trials that came up, as Federico already mentioned about the gene therapy trial for hemophilia B with SPARK, and of course the biomarin study, where I was particularly involved in for factor VIII. And as you know, and I think we have discussed the AAV-mediated gene therapy to death, where we know that the gene of interest is put in the vector, which is the AAV capsid, and we infuse it into the patient, goes into the liver, goes into the nucleus of the liver cells, and then starts to produce the protein in time. So we are really moving goalposts with gene therapy, and of course, starting off with gene therapy trials. So as I said, I think it is important. Patient engagement is very critical and needs significant thought, particularly in a country such as ours. And what I think should happen is patient information sheets should be co-developed with patients, or a group, or at least a group of patients in that particular disease area, because our country has very different levels of understanding and different cultural backgrounds. Education levels vary from rural to urban areas, and it is difficult to talk about genes and genetic engineering or capsid engineering, talking about the vectors, etc. So I think it's very, very important that you have the information sheets are actually tailored to our patients across the country. It's also important to understand that comprehensive care in inherited diseases or chronic diseases is not the same that I know of in the UK, for example. And the same, in the UK, the patients, I would have seen the patients for 20 years in their lifetime of their hemophilia care. It is quite unlikely that the same doctor would be seeing their patients for their lifetime. So it's important to understand the patient-doctor rapport, and it's very, very important for clinical trials and to make sure that you're consenting appropriately. And managing expectations is also very important whilst you take consent, that they don't believe that it is going to be a one injection and that's all, one and done. It is not that. So going to site readiness. So what is important in terms of site readiness? I think, sorry, sorry, can I just hide the floating controls? Yeah. I think it's very important to have a good subject expert as principal investigators. That's clearly a given. And there should be good governance and oversight that is required, research governance I'm talking about. What is also important is an excellent local laboratory, because many of the times you need the test to be returned to you very urgently, and you may have to take decisions soon, for example, to give steroids or to give another immunosuppressive agent when you think that the liver enzymes are going up. Dr. Jotwani talked about institutional biosafety committee. We need to have an institutional biosafety committee within the institution where these clinical trials are going to take part, where the trials are going to be held, and a very responsible research and development department in the institution, and a knowledgeable and responsive ethics committee is also essential. Persis mentioned about advanced therapeutics, IMP pharmacist, and I think we need to have a proper ATIMP pharmacist in order to make sure that the product is kept in the right temperature and it's received appropriately and can be given to the patient or made up properly so that it's given to the patient in the appropriate way. Again, there should be adequate SOPs and governance structure, appropriate biosafety cabinet to handle the product, and a dedicated trials unit with phase 1 capabilities. Inadequate refrigeration and freezer capacities are given. For cellular therapies or ex vivo gene therapy, we may require the bone marrow transplant unit requirements as is available in the country. What is more important is that we could improve the patient access by hub and spoke model, where this is routinely what is done in the Western world, where the gene therapy infusion is actually given at a hub, and then follow-up is done at a local site with appropriate oversight from the hub. So governmental agencies, and I think Geeta Jotwani has already spoken a lot about regulatory framework. India is joining the gene and cell therapy party a bit late. So what we need is adequate governance and fast tracking of clinical trials. Most phase 1-2 gene therapy trials will have a readout at about 2 years and continue to 5 to 7 years, and will have registry data thereon. Phase 3 trials usually start 2 to 3 years following the start of the phase 1-2, and licensing application at least after a 3-year readout from the phase 3. So it is going to take a very, very long time before, from the start of a phase 1 gene therapy until licensing. The safety of the patient remains the main concern, and of course, long-term safety is still a big unknown. And it is important to think about a single gene therapy registry that may be mandated for all trials, so that late adverse events can be compiled and learning shared among experts. Maybe utopian, but I think it is still worth thinking about having a single gene therapy registry. The preclinical data should be looked upon with a tooth comb, and I'm sure the guidelines already exist, as already talked about. The life sciences industry in India. It is called the pharmacy of the world. India has a number of companies making biosimilar antibodies, which are marketed across the globe. And I believe, maybe I'm a bit naive, that gene therapy or vector production is a step up from antibody production, maybe a significant step up, but still is just a step up from antibody production. The capacity and capabilities exist in our life sciences industry, and I'm sure we'll bring about cost-effective production with the scaling up of manufacturing. And it can certainly serve the Indian population and many more LMICs. I certainly know of 2 companies, I mean, there may be many more, of companies who are intending to set up commercial manufacturing initially for clinical trials, but of course going on to commercial manufacturing for gene therapy. And then finally, the science behind gene therapy. What we really need is to have many more medical colleges, to have a link to universities with biosciences and bioengineering departments, good library and access to current scientific journals, attendance into international meetings dedicated to science. I believe the clinicians and scientists need to work together very, very closely. Scientists should be part of specialty departments. What we also need is clinician scientists, like the likes of Persis and the likes of Vikram Matthews, hence more MD, PhD programs. The government should encourage such collaboration and provide funding to collaborative groups and a focus on gene and cell therapy research and academic clinical trials as well. We really need to play catch up with the new technologies compared to the rest of the world. I think I'm going to stop there. As I said, it's not a very scientific pitch that I had. So thank you.
Thank you, Dr. Savita, for this wonderful talk. We'll take questions at the end of this session. And so we'll move on to the next speaker. Our next speaker is Professor Vinod K. Paul. Professor Paul is a clinician scientist and a full-time member of the National Institute of Transforming India, acronymed as Niti Aayog. He heads the Health, Human Resource Development, and Nutrition verticals in Niti Aayog. More recently, he has been championing the Union government's core team for COVID-19 pandemic response. Professor Paul was a faculty at the All India Institute of Medical Sciences in New Delhi for over 3 decades, and also the head of the Department of Pediatrics for several years. Apart from being an eminent neonatologist, Dr. Paul has contributed to India's national programs on reproductive, child health, and nutrition. He holds positions in multiple international fora on child health, has been the co-chair of on the board of Global Partnership for Maternal, Newborn, and Child Health, and also a member of the United Nations Millennium Project Task Force on Child Health and Maternal Health. Without further ado, I now request Professor Paul to deliver his views on emerging therapies in India, the government priorities, and the way forward.
Thank you very much, Mr. Rao. Am I audible?
Yes, Professor Paul.
Thank you very much. Thank you for this opportunity, and thank you for engaging with the perspective on government's role and government's connect with the developments in this frontline area of gene-related therapies. Well, truly, I heard Savita talking about the significant potential that this nation has in the area of new therapeutics, not just potentially in the old therapeutics for which we are known as the primacy of the world. And in recent past, we have also seen that our vaccine enterprise can rise to the occasion, not only for India, but also eventually and even now for the world. So rapid development of several vaccines, some already in operations, and a few more, 4 to 5 more are right out there at the cusp of being used in national and global programs. And we do believe that the area, advanced areas, such as the gene-based therapies, cell-based therapies, is also a clear priority. And we have taken some important steps. A lot more needs to be done. We'll be delighted to be guided by a group like yours. But just to put the perspective, every year billions of Indians are affected by inherited monogenic or complex diseases. And we truly have very limited and very often no treatment options. A number of these disorders are associated very disturbing, very demoralizing lifelong disability. And of course, They are a significant burden to the family in more than one way, but also a true burden, economic burden to the society and to the system. A large burden of monogenic diseases in India particularly includes hemophilia, thalassemia, sickle cell disease, which I believe must be absolutely top priority because it afflicts our brethren and sisters in the tribal hemophilia. Very poor people, very, very poor people. Of course, muscular dystrophies, retinitis pigmentosa, primary immune deficiencies, lysosomal storage disorders, Pompe disease, and so on, cystic fibrosis. These are our challenges, huge, huge challenges. And if being one of the most populous nations in the world, our numbers are staggering. Just to take a few examples, hemophilia could be around 50,000 patients. Often we may not be detecting them, diagnosing them. 150,000 sickle cell individuals or more. 500,000 Duchenne muscular dystrophy. And retinal dystrophies to the tune of 1 in 4,000. If you add these numbers, perhaps they will run into maybe 70 to 100 million individuals all the time. So while we work on preventive approaches, counseling approaches, there cannot be a better way to address than to go at the root of the cause, to change the switch that controls these diseases. Gene therapy is a process of introduction, removal, or change in the content of an individual's genetic material with the goal of treating a disease and the possibility of achieving a long-term, sustained, full cure. That's the aspiration. We are mindful of the fact that there are significant developments in the world, whether it leads to gene therapy for retinitis pigmentosa or advances in hemophilia, which is the direction, or CAR T-cell therapy for lymphoma. And yes, India would aspire to be in the forefront of finding these cures and in the forefront of creating large stockpiles of these cures through our research and development and manufacturing capabilities. That's our aspiration. And we would like to realize this with the support of global community and scientists within our organization. We are working in that direction. In the next few minutes, I'll give you a little glimpse of how a few important steps are being taken. In 2019, we released the national guidelines for gene therapy product development and clinical trials. My team at Niti Aayog also facilitated this effort. This was very important for us to have updated guidelines, most fresh guidelines, so that there is a framework available. Without such a framework, there is no systematic trial, and then the nuances of specialized areas of research get lost in the generic. So this has been an effort steered by the Indian Council of Medical Research, but supported and aided and facilitated and participated by the Department of Biotechnology and the Ministry of Health and Family Welfare. Those who are hearing these terminologies for the first time, let me just say the Indian Council of Medical Research is health biomedical research system. We have a separate department in our science ministry, unlike the Indian Council of Medical Research, which is in the Ministry of Health. In the science ministry, we have Department of Biotechnology, which undertakes research in the areas of vaccine development, biopharmaceutical products. And therefore, this paradigm is somewhat divided, but we work together. So the upstream work would happen and be catalyzed by the Department of Biotechnology, including facilitating efforts in the industry sector that generally falls in the rubric of DBT, Department of Biotechnology under the Science Department, Science Ministry, and clinical trials, field trials, practice, evidence-driven practice of such therapies would fall in the domain of Indian Council of Medical Research. And then, of course, we have the Drugs Controller General of India system available to us for regulatory processes. So we have these national guidelines for gene therapy product development and clinical trials, 2019, which guide the journey of research in this area. And we define gene therapy products as an entity which includes a nucleic acid component being delivered by various means for therapeutic benefit to patients. These guidelines provide general principles for developing gene therapy products for any human ailment and provide the framework for human clinical trials, which must follow established and advanced principles of biomedical research on humans. We also commit that we will revise these guidelines, and we created a system within these guidelines to continuously refine them and revise them. I want to emphasize here that as per the clinical trial rules that exist today, the gene therapy products are what we call as new drugs, and they'll be deemed to be new drugs in time to come. And what it means is that this will be, the trials in this area would be treated as trials for regulation and not academic trials where we take a softer approach, not a trial on let's say, on remdesivir in an academic setting, so to say, by academics. So this will be seen through a, shall we say, in some sense, more robust system of oversight. More recently, the ICMR, the Council of Medical Research, has invited proposals for gene therapy research projects in areas of Duchenne muscular dystrophy, SMA, myopathies, retinal and corneal disorders, heart diseases, that is familial cardiomyopathy, generalopathies, and thalassemia, sickle cell disease, and hemophilia, and some of the priority areas. And also for traumatic therapies in the potential therapies in the areas of diabetes and neurological disorders. So we have a framework, we have some initiative being lined up, and a lot more can be built on this part of our effort. We also have been making advances in the area of stem cell research and its application. Stem cells have found use in leukemia, thalassemia, plastic anemia, sickle cell disease, and perhaps not in other areas such as stroke, traumatic brain injury, spinal disorders, muscular disorders, and so on. You know that better than me, where it works and where it does not work. But from our point of view, we have divided a clear-cut line as to where stem cell therapy can be given and where it cannot be given. It's been growing, in a sense, significantly. I would like this to grow much more. Couple of centers of excellence, a few centers of excellence in the private sector provide stem cell therapy. Stem cells and their derivatives fall under the definition of drug and under our law, and characterized as investigational new drug and investigational new entity when used for clinical application. Thus, as you would expect, strict ethical and regulatory guidelines have to be followed. The current landscape of stem cell therapy and research is guided again by another set of guidelines, namely the National Guideline for Stem Cell Research 2017. Again, ICMR and DBT and others joined hands to have these guidelines. They categorize stem cell research on the basis of ethical and safety concerns regarding the source of stem cells and levels of manipulation, which warrant iteratively additional review and monitoring depending on the type of stem cells. So we provide for permissible areas of research where it encompasses in vitro studies using stem cells isolated from tissues can be done with a prior approval of Institutional Committee on Stem Cell Research. So there's a system where you need to apply for. There are some restrictive areas of research where approval is taken at a different level, a higher level. And then there are prohibited areas of research in stem cell, according to the current state of our framework. It includes, for instance, research related to human germline gene therapy and reproductive cloning. It includes clinical trials involving genogenic cells or use of genome-modified human embryo, germline stem cells, or gametes for developmental propagation. So this is all there in the public domain. These guidelines are very important, are also providing framework for umbilical blood banking and storage, transfer and transportation and storage of stem cells, practical guidelines, the standards in a way. And it makes it very clear that only legal use of, only the accepted use of stem cells is in the domain of using hematopoietic stem cell therapy in hematological, essentially hematological and some non-hematological conditions. And anything outside this is a clinical trial. It has to follow the rubric, the guidelines, and the laws and regulations related to clinical trial. So the conditions where it can be used, as you would expect, would be acute myeloid leukemia, acute lymphocytic leukemia, CML, germ cell tumors, aplastic anemia, Fanconi, thalassemia major, JRA, systemic sclerosis, and so on. And this list is available. Other than this list as an annex available to these guidelines, everything else is a clinical trial. Although we are aware that there are people who are using stem cells in some of the therapies that are not listed in that sense. But for us, that's outside the framework and therefore should be undertaken only in research mode. So except for hematopoietic stem cell transplantation, for hematological and the disorder that I said, everything else is a clinical trials, and therefore those trials have to be conducted with clinical grade cells, good laboratory practices, good manufacturing practices, and good clinical practice guidelines. There are two examples of such work being done. One with Bangalore-based, two examples I'm giving. InStem is an organization which is working with the Christian Medical College, Bangalore, is conducting a large study, a large trial on stem cell therapy in sickle cell disease. LV Prasad Institution in Hyderabad has been conducting stem cell therapy in eye conditions, and these are ongoing clinical trials. That brings me to the next category of therapy, namely the chimeric antigen receptor T cell therapy, CAR T cells. These are genetically engineered to produce artificial T cell receptor, which is widely used to develop in developed nations, developed laboratory, shall we say, for immunotherapy, for cancer in particular, you know, again, better than us. We are mindful that CAR T cell therapy costs a fortune, lifetime earning of perhaps 100,000 people or more. And therefore, India, for this to be used in India, we have no way, but as the Prime Minister exhorts us all the time to accept to be Atmanirbhar or self-reliant. That's the philosophy we have followed in many, many domains, including the recent vaccine journey that we have taken, Atmanirbhar, self-reliant. And India has done that, by the way, in many other fields, including defense, for instance. I'm pleased to inform you that an excellent team of researchers at the Indian Institute of Technology, Bombay or Mumbai, in collaboration with Tata Memorial Center, which is Tata Cancer Hospital, is conducting a first indigenous CAR T-cell therapy trial. This is an indigenously developed therapy, perhaps the first product that we are able to showcase and take to clinical trial. And we look forward to seeing the final outcome of the study. The study is being supported by the Department of Biotechnology. It uses lentiviral technology platform. The CAR therapy is being facilitated by the Department of Biotechnology. And we have an institution within DBT called BIRAC. It is Biotechnology Industry Research facilitation platform, an extension of the government, but works independently to an extent as a company, non-profit company, which is then able to make interface with academia, with industry, and the other ecosystem, startups and others, and able to take steps in translational work and lab to industry to patient work in a flexible way. And so BIRAC, I like those who are interested in working in India to connect with BIRAC, apart from DBT and ICMR and others. So this first in India gene therapy is being facilitated through the BIRAC system. And we have an overarching umbrella mission called the National Biopharma Mission, which is supporting the developments in this area, as well as in stem cell research that I mentioned. From the DBT side, at least these two and something more being supported. So lentiviral vector manufacturing facility is being supported. It is a GMP-enabled facility for T-cell transduction and expansion of CAR T-cell manufacturing. It will take to other organizations. And the development of this technology for diseases including ALL, multiple myeloma, glioblastoma, hepatocellular carcinoma, and type 2 diabetes is also in the offering through that system. So there is one definite product which is being tested on the ground. And then there is this seeding of research activities in other areas. The mission is also proposing to have a center of excellence for training to those who, and capacity building and mentoring, hand-holding of laboratories and individuals and industry, that we are building this at this moment, to be able to support, facilitate, and handhold this difficult and clearly exciting area of solution finding in human disease, human health. So friends, I have given you an oversight of how, specific oversight today, essentially, that the Government of India views this area to be a high priority, emerging therapies as a very high priority. We have taken steps in the form of guidelines, in particular clinical trial frameworks as required. So regulatory system has been adapted. We are encouraging industry, and we have exciting possibilities and excitement around the industry. And some of those names you are familiar with. We are happy to work with them. We would like to see India as the capital of such therapies going forward, purely because we need and we have the capacity to do so. We have taken some steps. We would love to take many, many more steps in this direction. We are also examining at NITI Aayog a research-linked incentive system for the industry, so that going forward, there is an enabling, very definite enabling provision for productivity-linked assistance, or shall we say, incentive in the form of taxation and other areas, apart from the funding opportunity that we would like to But that work is still ongoing. But clearly, we would like to press that button as well, because that then makes such effort attractive even to large companies and of course to startups as well. I'm excited to know that IRTAS and Reliance are also interested in using the convening mandate of my institution. I'll be happy to get the industry and the scientists in our country to be together. I told this to Dr. Rangarajan, except that Corona had occupied a lot of our workspace, mind space, bandwidth. But I would be very keen to look at how we can enhance this, bringing together the players, serving as a clue, finding out how to facilitate this effort and how to remain in step with developments in the world. I invite you to think about collaborations with others and scientific groups in the world. This is an area which is in the early stage of the rising curve. We believe we will stay with the development, with the dot as it moves up. We still have an opportunity. But above all, I would like to believe that if the world has to have such therapeutic solutions, perhaps India is the best bet. So let's join hands. Thank you very much, Jay.
Thank you, Professor Paul, for this inspiring words and also for outlining your vision to develop and deploy novel gene therapy products and cell therapy products for patient care in India. We sincerely appreciate your time. And now I request all the panel members, Dr. Federico Mingozzi, Dr. Savita, and Professor Paul to join the discussion. So the first question is for Dr. Federico Mingozzi. There are studies reporting that in case of Leber congenital amaurosis type 2, the effect of RPE65 gene therapy product declines after 3 to 4 years. What factors are responsible for this decline? And what kind of changes do you think in the current regimen should help in order to overcome this problem?
Thank you. This is an excellent and very important question, which is the one about durability of gene therapy. In the case of the studies that I believe the person asking the question was referring to, they were published a few years ago. And I have to say, the first comment is that we do not see this loss of let's say therapeutic efficacy over time in our patient population from the older studies, the phase 1, 2, and phase 3 trials. However, there are possible factors that can lead to loss of efficacy, which is the fact that when you inject a vector subretinally, you don't really cover the entire retina. And so areas of the retina may continue to be affected by the disease and degenerate, and that may affect the overall outcome of vision in a subject. So it's hard, we're talking about an eye disease where the endpoints are complex, and so it's hard to really pinpoint to one specific factors, but one possibility is that the efficacy was low to begin with, and then the disease kept progressing.
Thank you. Thank you.
To improve it, of course, is to have a better targeting of the entire retina at a higher level, right? That would be the way to improve it, to improve the outcome over time.
Right. Thank you. Thank you, Dr. Mingozzi. The next question is for Dr. Savita. In India, how difficult would it be to get patients participate for clinical trials for gene therapy? Is there enough awareness among the people? And what has been done to improve this awareness?
Now, I can talk certainly about hemophilia because I have been interacting with a lot of patients for hemophilia. Now, I actually undertook a seroprevalence study in the hemophilia population, and I have interacted with all of them. before taking samples, just the blood samples for seroprevalence. And in fact, for doing seroprevalence, I had to discuss what gene therapy actually meant before telling them about AAV vectors. So this is what I commented on, that the science, knowledge of science is very varied in our population, and therefore it takes time to talk to them about what gene therapy is, and you have to talk to them in a layman's language. But I do believe that most of the patients do understand what I'm talking about. And not only that, and I think there will be a lot of individuals who will definitely be interested in gene therapy, not necessarily for the fact that they are vulnerable and don't have access to proper care, but I think genuinely interested in the science as well.
Thank you, Dr. Savita. My next question is for Professor Vinod Paul. Given the immense response generated for COVID-19 by the mission mode activity by the government, can the government initiate such an initiative for gene and cell therapy? For
well, the
treatment
two
of
things are, thank you so much. Firstly, we must appreciate that the science and technology endeavor for COVID-19 had mostly a familiar territory. Whether for India it was, vaccine, including a vaccine, or ventilators, or PPEs, or even some drugs, it was a familiar territory. The gene and cell-based therapies are very advanced, very niche product, but that's the fun, because if we have to move into future, then this is time to catch them. So I think it's a mixed aspiration, both. The short answer is that we recognize this area to be so very important for India's leadership, for India's own health of India's people, and also India's aspiration to be in the forefront for SNT and for medical products. Yes, that kind of a mission mode activity can be mounted. Yes, it is possible. But the situation right now where we are, I would like to the stakeholders to come together. And ultimately, I think it is about excitement around the industry. The people who bring this enterprise together, and if they, because remember, Pharmacy of India runs on the industry. R&D enterprise is there. But I think surely I would like this to be a mission mode work. I like us to zero on to priority areas and find solutions where the best bets and low-hanging fruits are. We would like to see coming together of a critical set of stakeholders. So yes, the short answer is yes, it can be done. It is difficult. Nonetheless, we would like to work in this area, and we have done the critical elements. You must appreciate that if there are no guidelines for research, how do you go? Then it gets mixed up with treatment for malaria. This is so nuanced, and the researcher is very unsure what is legal, what is not legal, and there are so many deep ethical issues. So that part has been cleaned up with the two specific guidelines that I have referred today, and we all work together in that area. Some of the people who are attending have also been a part of it. Thank you.
Thank you, Professor Paul. The next question is to Dr. Mingozi. It's a two-part question. Could you please suggest the areas in manufacturing where low-income countries should focus on to reduce the cost of production? I'll come to my second question after this. Yeah.
Well, I mean, that's a very interesting question, because of course what drives the cost of goods is at least for my knowledge of in the field of gene therapy, is part of it is the cost of raw materials, and then it's the scale. So there's a lot of technological innovation that needs to go into the scaling up gene therapies. And I'm talking about for in vivo gene therapy, right? And then for ex vivo, there's a sort of other challenges. And then for raw materials, again, it's a matter of probably in some cases the scale and the availability, because one of the let's say one of the components of the gene therapies is having like plasmid DNA plasmid made at large scale, and that's still a very limiting for the field, and of course availability drives prices, right? And so probably probably the scale up is the most important part, I would say, in not being exactly a super expert in that, but I would say, and how this country could contribute is probably there's an opportunity there to invest in manufacturing in these countries, where I would say you probably can access highly skilled labor, and the labor is another part, right? That is hard to source, and it's also a cost driver. And so I would say probably some investment in manufacturing will be at the end also possibly help gene therapies to happen in these countries, right? It's, as I say, it's a major barrier to entry, having availability of clinical grade vectors to perform trials. And so if there's a way to have them, to have access, I think they will be really promote a lot of local initiatives, right? Because this is probably what you want at the end of the day.
Thank you. And just a small follow-up question. And that is, are you conducting any clinical trials in LMICs currently?
The short answer is no, but we're looking into that. One of the factors that I mentioned in my slides is there's a layering of components, let's say. First is that at the moment we don't have phase 3 trials open, and we're working on it for some of our products. And the second is that the phase 1-2 trials are for gene therapy are still very complex, because there's some technology hurdles that I mentioned earlier. So we need to really make sure that we can follow subjects almost real time, and sometimes that is then is what the clinical team and the regulatory team feels like, is that they want to have like very close the close monitor the subject, so in clinical sites that are actually closer to us, or in situations where we're sure that there's a close connection with the clinical center. So I would say, and also the standard of care is all very, very uniform. And I think those are barrier to entry that can be addressed. I think as I was following the talks, one thing that I sometimes feel like is that almost like companies, like biotech companies like us, need to be educated about the resources that we can access, and sort of reassured that we can actually do also early phase trials. I think that's probably a point that will be helpful. And again, I'm giving you the opinion of, I mean, I represent the science and the research spark, right? So, but I think somehow, I think sometimes it would be good to engage in a discussion and try to move things forward by exchanging information and really look at available resources.
Thank you. Thank you, Federico. My next question is for Dr. Savita. Do you anticipate that gene therapies will primarily be developed by companies based in India or by international pharmaceutical companies for deployment in patients?
Well, I clearly said that I know of certainly 2 companies who are already in the game of developing their own gene therapy products. And And I know that they are very keen on making it very cost effective. And the other thing that I just wanted to answer, a part of the answer that Federico gave was about, how companies like Spark do not know what is available and how capable we are. And in fact, I am going to have a talk with them next week. So I hope I can actually make him understand what the possibilities are in India as well. So I think this is all about, I think the COVID world has told us how collaboration is important across the globe. There is no question that we all have to work together, whether it be a pandemic or whether it be how to treat diseases for the future. So I do believe that we will be talking about both indigenous as well as global products.
Thank you, Dr. Savita. So the final question for this session is for Professor Vinod Paul. There is a gap between scientific research and common man understanding of science. How can we bridge this gap?
Communication. Simplify the concepts, reach out to people, use channels, multiple channels. The outreach of channels depends on social strata, gender, location, and so on and so forth. But yes, it is very, very important. And in the context of COVID, you may have seen, at least in the middle class space, so much of discussion on doses and side effects and duration of dose, DNA, mRNA, all that. Yes, we are accountable to the society to be able to explain the scientific basis of what we offer as treatments, what we offer as therapies, what we convey as simple messages, even why masks should be worn, why home care of COVID-19 patients has to be according to a protocol, for example. So I think when you lace your messaging with scientific information, clearly communicated, it's helpful. Very important, I have learned over this period of time, is the clarity and the confidence with which you convey the message. As scientists, we are very often traversing the domain as a gray zone domain. And we know that we have uncertainties around what we believe. There is a 95% confidence interval around whatever we believe is the truth. But then that call must be taken in our mind before we open our conversation. Because what we should convey should be in the best interest, a sincere interest of the target audience, and it should come through as clear cut. People expect scientists and doctors to give a clear information, particularly when it deals with, shall we say, public health. Yes, in individual decision making, whether to have pregnancy or not to have pregnancy in a certain setting, yes, indeed, there are balances to be communicated. But for mainstream messages, yes, I think clarity and the tone and tenor of sincerity is very important, and using different channels to reach out to the local communication system and language. Thank you.
Thank you, Professor Paul. True words. I think these would be very effective in communicating our advancements and to reach the common man. Yeah, very true. So we come to the end of this panel discussion as well as today's meeting. And I'm going to give a few closing remarks. First of all, I'm sure you all agree that we had a very enriching and fascinating meeting today. It highlighted the status of cell and gene therapy in both India as well as UK, regulatory perspectives from Indian Council of Medical Research, the industry perspectives, and also the governmental priorities and ways forward to take up this field so that it benefits the patients. We hope that this will stimulate and further discussions among different stakeholders so as to start new clinical trials in India, development of new gene therapy products, and ultimately make use of this novel mode of therapy. I wish to thank all the speakers and also the audience for their enthusiastic participation and engaging questions. I also wish to thank our sponsors, Spark Therapeutics, in collaboration with Cure SMA India, the Hemophilia Society, Mumbai Lucknow chapters, Indian Society for Clinical Research, Somaya Ayurvihar, and Organization for Rare Diseases in India. In addition, I would also like to thank the American Society for Gene and Cell Therapy and the Global Outreach Committee, the chair, particularly Dr. Kenneth Cornetta, staff members, Samantha K, Betsy, Caitlin, and Sarah for the preparatory work. In fact, this preparation started almost 9 months ago. So it was due to their untiring efforts that we could see this meeting today. And finally, I would also like to mention a feedback form would be circulated by ASGCT to all the participants on this meeting. Please give your feedback. We would like to improve in the future meetings. And for those of you who need more information, you can also refer to the patient education site at patienteducationasgct.org for additional information on ASGCT's initiatives. Finally, I also would like to thank Professor Vinod K. Paul. He's busy. He is in the middle of pandemic response. He found time for us, inspired us with his views, and we look forward to taking it forward, sir. With this, I thank you all once again, and I have a great day ahead. Thank you, everyone.
Thank you. And thank you, Dr. Paul, again, once more from me as well. Thank you. Thank you.
Thank you.
Thank you, everybody. Jaya Savita, thank you so much. I hope the ASGCT staff have some coffee now to keep going. And the speakers, you guys put together a wonderful program. Thank you so much.
Yeah.
Thank you, Ken.
Thank you, Ken.